Hello, everyone. Just trying to preempt any problems. Not any problems, the problems I'm going to have. Well, hello, Brian. Hello, Alt Miss Frizzle. Also, I hope you're ninth grade teacher. I thought you said that and then I forgot. But look, your little shout out works. Yes, we're just waiting for James to raid us, I guess, and join me on this call, which is going to completely mess up the camera setup. So that's going to take a second to figure out. Probably more than a second. Fossabot? Okay. This is the day you die. Fossabot speaks too often. I need to shut it off. Uh, let's set this at 75 minutes. Save that. This one's 90 minutes. Save that. All right. All right. So I hope that I hope that keeps. Oh, shoot. And Fossabot decided to speak a second time while I was looking away. Yeah. All right. How's your Wednesday? How are your Wednesdays? Yeah, far too chatty. The way I thought I set it up was that it's only supposed to speak every like hour and only if uh, there actually was a certain number of messages. Like if it's, a Sunday, no one's watching, I don't want to just spam me and only me, but it does. You're good. You're writing all day, so this is a much needed break. Well, I'm glad. And Alt Miss Frizzle, you got to move stuff into your new house. Awesome. Oh, and we just got rated. So I'll wait just a second. I personally get ads every time I join a new channel, or a new channel. So maybe no one can hear me right now. Okay, I'm getting some hellos. So, hello Raiders, welcome, welcome. Oh, awesome. Okay, thank you, Genum. I feel like every time I am a part of a raid and go to a new channel, I don't I get an ad immediately. Maybe it's just my luck. Uh, hello Anima Confusa. Hello Sci-Fi Gal 13. Hello Gopher Fluffernut. Hello Paperclips. Nice. I'm just waiting on James to join the call. I hope I sent the right link. Let's see. Yep. Oh. And he's rating. Go back. Go back. Don't show that. Rating with a group of one. Welcome again, I guess. There you go. Okay. Struggling, but you're here. All right. When you get the chance, try and join me on Discord. Meanwhile, I got to move around some things. I see that the chat is going to overlay where it shouldn't be. Maybe this is just a bit too big. Scoot that over. Scoot you over. All right. He's pulled another Hewlett. Pulled a Hewlett with the raid function. I haven't been on a Tech Bandit stream late enough to join a raid. He's managed to raid with David and from his own channel, lol. Zelenkadet. Yeah. Well, welcome everybody. I'm glad you could hear me almost immediately. Once we get the second, the guest here, and then figure everything out, because I know I'm going to absolutely mess it up. You're clicking join guest chat and not much is happening. Oh, okay. Let me do, what should I do? That's annoying. Are you in my server? And then go down to voice channels. There should be something called general and then something called guest chat. Is that what you're trying? Ah, okay. Well, change of plan. 1 second. Let's see. Get rid of that. I'm disappearing myself just for a moment.
There we go.
Hello.
How's it going?
Good. Hold on. Now I have to figure out how to fix this. Okay. Got one window capture. Also, I can hear you, so that's good.
That's a handy thing, yeah. I don't know what happened to some of my capture stuff all fell apart today, so like even Discord had like reverted to use something else I've never seen before. It's like my capture card decided to call itself something new.
Oh, that's annoying. I every time I've had a guest on, it's like I have to completely reconfigure the video every single time. That just what it wants to capture is never the same thing. Okay. Speaking of which, I have to change some things really quick. Okay. All right. I don't know if you're watching stream, but I'm slowly getting you on screen.
Slowly turning up. It's all right.
Hey, Brian, can you hear James? Do you mind if I call you James? Please,
yeah, James, fine.
Please call me Marcus.
Sure.
Dr. N is the partially anonymized, but not actually all that difficult to like find me sort of name I chose for streaming.
That's from academia. Like,
yeah.
I realized this early in the days of YouTube. It's like I can't really, if I ever show my face or anything identifiable, it doesn't matter what pseudonym I use. People are gonna work out who I am.
Yeah. It's just it's what's gonna happen. So I sent you the link for this, just in case you want to like at any point just drop something in. That's kind of what it was for. Behind the scenes, everybody, I usually try to make a slideshow. This one is only about an hour old at this point. It's been quite the week. And all it is to sort of cut down on the amount of Googling for images, because as much as fun as it is to just talk while streaming, I mean, it's a semi-visual medium, right? It's nice to see people. Yeah, so welcome. Also, thank you for coming.
Oh, my pleasure. Absolute pleasure.
Yeah. So I don't normally introduce myself usually, but I might as well. although I feel like I did because I was just on someone else's stream who did a whole interview. So it felt like I told my entire life story. Uh, so I'm a postdoc in a state not too far north from where you are because you're in Georgia, right?
That's right.
Um, yeah, I'll call myself a molecular biologist, a genomicist. I don't really know. I at some point looked up. I saw that you called yourself a microbiologist. I don't know if you count that. Like my degree's in biochemistry. I don't think of myself as a biochemist. Um, yeah.
Yeah, I'm definitely a microbiologist. It's yeah. There's that weird line with biochemistry and like people who hate chemistry but still are technically biochemists. Like if you do anything that isn't like, oh, if it's not inside a living cell, it's biochemistry. But yeah, it's I could slide into being called a biochemist and still do pretty rudimentary chemistry.
I mean, you've done a Western blot, right?
Yeah,
You're a
that's,
biochemist.
that's
Congratulations.
it. Yeah, that's how it works.
Yeah, no, exactly. I mean, sort of the same. I don't want to think about pKa's or like binding coefficients at all. I was forced to take, thermodynamics,
Mhm.
pchem, basically. Don't want to think about any of that. I am much more into like the genome, all the squishy little things bumping into each other and maybe doing something.
As as I've got further in my career, though, I realized, much to my chagrin, how important biochemistry actually is. Like,
Yeah.
there's a lot of things like, ah, crap, I really should have just paid more attention to that. There's a whole bunch of metabolism stuff I get up to, and it's
Ooh.
like I've had to relearn a bunch of stuff, which
Yeah.
I was taught at some point a decade and a half ago now, but yeah.
Absolutely. Teaching a group of students, even just glycolysis this past semester, and pretending like I remembered all of this just off the top of my head was a little bit of a challenge. Um,
The the classics, the Simpsons, where they're saying, "Oh, it's a Family Guy." I think it might be Family Guy. They're like, "Oh, you just teach people how to play piano. Just take the course yourself and be as long as you're one lesson in front of the kids, you're fine."
oh, yeah.
It's some chemistry stuff I've taught is like, yeah, very much like I wasn't just reading this the day before. What are you chatting about?
Yeah. For anyone out there who is a maybe someday wants to be an educator, that is absolutely true. The program that I'm in has a focus in teaching and research.
Oh, yeah.
And we were told all the time, like, do not try to set yourself up even a week ahead of time, maybe not even a day ahead of time. Just stay a couple pages ahead of everybody and you'll be fine. It's so true. Yeah. Also, Alt Miss Frizzle, yes, we should talk about T. magnifica sometime. Have you heard about this? Um,
Is it the eyelash size bacteria?
yeah, I didn't.
I mentioned to you on.
Oh
Yeah,
yeah, you
I sent
did.
it to
You
you on
told me
Sunday.
about it.
Yeah, yeah, yeah. It's
Oh
like,
my gosh. See, I said it. I said it's been a week on your stream. I can't keep things straight. All right.
so I mean, so for the audience, it's a bacteria that was discovered. I think it was discovered 2009, and they only just like now got around to formally identifying what it is. But it's macroscopic in scale, which makes sense for the stuff some of the stuff we're going to talk about today. It's huge. Like you can see it with your visible, like naked eye. Which for a bacterium is kind of, well, very unusual.
Yeah.
There's some really cool, again, really cool biochemistry stuff there where they've got like
They,
separated ribosomes and stuff, almost like organelles.
yeah, yeah, and they the thing I'm interested in, they keep their DNA separated into little
Mm,
compartments, which
yeah.
bacteria they will organize where their DNA is sometimes, but the way it sounded, I honestly didn't have access at the time to look at the article, and then since haven't looked back on it. But I'm very
As long as you read the
curious.
abstract, you're fine.
Yeah, exactly. Okay, I here's a picture of it. So next to a coin. I'm sorry it's such bad resolution for everyone on stream. When you told me about this, only via chat, obviously, I can believe a bacteria that's very long, like as long as an eyelash. I wasn't expecting the diameter to be something that we could actually see. So these very pixelated squiggles here, those are apparently single bacterium. So very
Which is
cool.
nuts. Like,
Yeah.
just, like you could fit, just guessing, but on one of the little bas reliefs of that coin, you could probably fit a million bacteria normally. Like like it's we're talking 6 micrometers in length. So having something that's centimeters up to centimeters is just phenomenal.
Yeah. Yeah, I definitely want to learn some more about
Millionfold.
this.
Yeah.
Yeah. So, Alt Frizzle, thank you for reminding me of that. Yeah. If not really necessarily the topic of stream today, but honestly, future thing that I'm definitely going to have to mention. Oh, is there a way to turn James up a little?
I can lean in or
Nope. I got it. I got it.
okay.
Okay. That should, for everyone on stream, have bumped it up a little bit. But
I
yeah.
can keep talking and see how that feels.
And I know you were just on your stream and we're talking a
Yeah.
little bit about yourself, but I interrupted you also. But would you mind just introducing yourself a little bit?
Sure. Sure. So I'm James Gurney. I am a researcher in the phage field, so bacteriophage therapy research mainly. I work at Georgia Tech currently, that's in Atlanta, and I'm about to start my own lab at Georgia State in 6 weeks.
Wow.
Like, any day now. It feels like it both feels like tomorrow and never at this point, because it's like, oh, it's like Christmas. It's just gonna arrive, and it's also terrifying. So it's like, oh God, please don't come. So yeah, my lab is going to be working on co-evolutionary dynamics between bacteriophage and their bacterial hosts. So my PhD was unusual in the sense that it was very much microbiology, like with a focus on disease and treatment and antibiotic resistance, but also coming from a social evolution side of things. So how do bacteria use quorum sensing, the ability of bacteria to communicate? So bacteria aren't mindless to their environment or the population around them. They negotiate and plan and strategize depending on the environment. One of the ways they do this is by releasing small signal molecules, so little tiny cheap chemicals into the environment. These build up as almost like a census, right? So the more of these molecules that are around, the more bacteria, give or take. And they use this to plan: are they going to make the genes that make you sick? Are they going to make the things that turn on biofilms, the things that make mucus and all sorts of cool things? But the way we tackled these questions was from a social evolution question. Like it's difficult, not impossible. It's difficult with a naive understanding of evolution to say, well, how does this trait evolve and be maintained? I just noticed the lights are going off.
That was someone
How
just
does this?
followed.
Okay,
Thank you, Paperclips, for following. That's that. Yeah. Just
I'm
ignore
not going to
that.
turn that on. If I remember, I'll tell you about my fiasco of my lights at one point. So there's an evolution question here. Like, if you are a bacteria or any organism, and you make something called a public good. So let's say I make an enzyme that breaks down a carbon source in the environment. Bacteria don't have hands, they don't have mouths. They pump these enzymes outside the cell. It breaks down the carbon, and that carbon will freely diffuse away. There's no guarantee that carbon, the breakdown product, the thing you want to eat, will diffuse back to the bacteria that made the protein. So it becomes a problem of sociality. If there's another bacteria in the environment that isn't making that protein, it now gets a free lunch without investing the energy to make that protein. So this now becomes a social evolution question. And so my supervisor at the time was one of the original people in this field of social microbiology and had done some really cool work. And we carried that on by asking like in-depth questions of like, how do these signal systems evolve? How do they change over time? What are the limitations of them? And how do if we play around with the system, how do the bacteria respond to that? Because a common idea was like, well, here's a potential therapeutic target. If we knock out these communication systems, the bacteria are going to be less deadly. What actually happens if we do that, right? That's very naive. You're like, oh, they'll be fine. Nothing will ever change ever again. It's like, well, they're probably going to change. They're probably going to evolve in some way around that. And a lot of my papers from my PhD were looking at these kind of questions. And from there, I went on to work on phage therapy. Since I was a kid, I've been like obsessed with phage. It's was an odd child.
I feel
I
like anyone who's gone through their PhD, postdoc, or entering
yeah,
a professorship was a little bit of a strange
I think
child
it's
at one
common.
point. It's pretty common.
So the thing that got me really interested was my mom was bringing me home from like after school club. So because she was working night, she would go and she'd leave me at the daycare thing afterwards. And one of the kids was sick, and we were talking about how. So we're talking like we're academics at the point. There were our pipes. The kid, myself and Charles were discussing. We were talking about, I asked my mom on the way home, like, why don't they treat disease with diseases? Like, if you're coming from that position of like a naive child, like you have an army, another army, you can make them fight each other. Why doesn't that work? Why don't diseases attack each other? And then my mom was like, this is she has a degree in microbiology, so she was like, oh, I have just the thing for you. So she bought me this book that had, I'm sure she had it already in the cupboard because she was like that. One of the it was a book of like different animals, but mechanical. So instead of like
Huh.
flesh and bones, it was showing you how like the biological systems worked if they were to be made in machines. So like there's a crocodile, and it showed you how the mouth would snap shut with springs and levers and stuff. And it's very much, my dad was an engineer, my mom was a biologist, so it's like very much perfect book for their child. But one of the pages at the back was this phage, and this is like early 90s, and the book was a little older than that, so it still had like punch card DNA instructions. So the DNA was shown as like punch card things running through this phage. And from that point, I just thought these were amazing. And literally on the back burner the entire time, my degree, my PhD, I had this idea near the end of my PhD about evolution stuff and phage, and realized there might be some phage that target efflux pumps. So one way bacteria develop resistance or maintain resistance to antibiotics is they have these pumps which will literally just pump the antibiotic out. I was actually thinking about it as from a social evolution point of view, that we were having these lots of discussions about efflux pumps and evolution. If you as a bacteria, there are two different ways you can get rid of an antibiotic. You can degrade it, or you can pump it out. If you degrade it, again, that's kind of like a public good. You're doing a benefit for the society. You're removing that toxin from the environment. If you pump it out, it's someone else's problem. So there's different co-evolution issues there, and different questions about how that sociality might evolve in different systems. And from there, I was thinking a lot about efflux pumps. And again, something in the back of my head phage and something connected up. And I was applying for jobs, and there was one in the south of France, in Montpellier, which I didn't actually know where Montpellier was. I just saw the job. I was like, oh, that seems cool. I should apply for that. Then I found out it was in France. They were looking at phage co-evolutionary dynamics. I was like, this is the perfect place for me to go. They're working on phage. I can take my idea with me, and we can work on it. I had a chat with the guy, and kind of dishearteningly, he was like, oh, that's a great idea. My collaborator Paul is already working on that.
Oh,
I was like, oh,
oh
god damn
no.
it. So that was like the second time that Paul Turner, who's at Yale, I am a collaborator with him. Like he was one of my supervisors for my fellowship. So it all worked out. But it
Yeah,
was
yeah.
like he'd also done, like, there's another social evolution thing like game theory and phages and viruses. Like I had this idea when I like a few weeks earlier or 2 weeks earlier than that, and I was like, oh, that'd be cool. I wonder if anyone's done that. And he'd done it like in the 80s. So I was like, god damn it. But I was catching up with him at least, so he hadn't found it yet. So he was working on this project of, are there phages that bind efflux pumps? And they found one. They isolated it from a lake, isolated it from a lake, and that phage has now gone in to be used in therapeutic processes. So I ran a bunch of animal experiments showing it worked over the treatment window, showed that it was effective under combined antibiotic therapy. And then it went into a bunch of cystic fibrosis patients and a aortic dissection patient. So quite old patient, his aorta basically erupted.
Oh.
They fixed it up. During the fixing process, Pseudomonas aeruginosa, a particularly nasty bacteria, got in there and started eating away at his chest cavity effectively.
Oh.
Highly antibiotic resistant. This phage targets Pseudomonas aeruginosa, so they were able to give it to him, and he survived.
Wow.
He's passed on now, but he was 85 10 years ago, so like it was
Yeah.
natural causes.
Still, it's amazing.
And yeah, so from there, I've continued working on ideas of how these phage and how these bacteria co-evolve together, how that may change the efficacy of these therapies.
Yeah.
So always coming from a co-evolution or evolution theory and applying it to medical approaches and molecular biology. That's my science history
Life.
in a nutshell
Yeah.
again.
Remind me if I'm wrong. Are
No, go ahead.
efflux pumps are actually pumps like ATP dependent, or do they depend on, like exchanging when one molecule goes in, one molecule goes out, or
They
are they active
some of them
transport?
some of them require ATP. I can't remember
Okay.
which family require ATP, but
So
there's
you can't see it,
um
I don't think, but I have an image up right now. So everyone who's
okay.
been watching this image that I had up there,
Yeah.
up here, these are different efflux proteins, none of which I know or recognize. I do not work in bacteria myself, but they're effectively proteins. They kind of are usually shaped like barrels, largely,
Yeah.
with a
Yeah.
channel through the middle that is the perfect size and shape to push out a molecule of some size.
Yeah.
And what you're saying, what I think is very clear, what you're saying then is right. They accidentally get exposed to some sort of uh, antibiotic, but they have the perfect pump that will just shoot it right back out when needed. So I'm looking at them. There's some pretty big ones too.
There's a whole bunch. So the ones
Yeah.
I work on are the MEX. So it's in Pseudomonas. So it's a MEX system, which is if you like the other name for them is a Toll C, which I think is like E. coli based. So
Yes.
they cross both. It's gram negative, so they cross both membranes.
There's.
And
Are you saying like MAC, like Mac?
MEX MEX.
Oh, okay. I see a Toll
Yeah,
C
but
on here. Hmm.
it's ABC superfamily.
Yes, that's
It's
what it says.
yeah.
ABC Superfamily. Um,
Okay, good.
okay. Also, I want to acknowledge Sci-Fi Gal asked how you even heard about phages as a kid. Sounds like that book.
Yeah.
So very clear there. Okay, that's awesome. That's really cool.
Yeah.
Also, yeah, a little bit of I feel like a lesson in there too for anyone who's in science or going into science, which is if you find someone working on what you want to work on, make them your advisor, your friend, or your
Yes.
collaborator.
Yeah. Don't make them your enemy.
Yeah, just don't make them your enemy.
Yeah, yeah. Paul's great. He's been very supportive. Again, I basically ambushed him at a conference. Like the second or third month I was in France, there was a phage conference back in London. So I flew back to the UK and was just basically trying to find him because I knew he was coming. I didn't really know what he looked like. I'd seen a picture of him, but I was like, I never met him. So I saw him come out of the elevator as we were just leaving for the day. So I pounced on him. I was like, "Oh, Paul! Like, I need to talk to you." So
And one
he's
more, I just have one more question,
yeah,
just because I'm very interested. And then I want you to like take a couple steps back, and we'll talk about what is a phage. But
yeah,
so are the phages? When I think of a phage or a virus, it's
yeah,
recognizing some surface protein that it
yeah.
that then allows it to invade the cell. So are these phages that are simply recognizing efflux pumps, or are they using them in some way? I guess I don't
Yeah.
understand.
We don't know. I don't think they're using them. I don't
Okay.
think they're using them to transport the DNA across. They're probably recognizing that structure on the
Yeah.
surface, and they have co-receptors as far as we can tell. So the efflux pumps seem to have something called the type 4 pili as a co-receptor, or LPS. So
Okay.
LPS is like the general structure on the outside of
Yeah.
of gram-negative bacteria. We don't think they use them to like it, even though it's a hole. Like it makes sense. Oh,
Yeah. Yeah.
hole in the cell, shoot your DNA through it.
It's,
We don't think that's the case, and
yeah.
part of the evidence for that is another common mutation that allows bacteria to develop resistance to these phages are things like ton B, which sits on the inner surface. So there's two membranes,
Yeah.
outer membrane and inner membrane in gram-negative bacteria, and then like peptidoglycan in the middle.
Which we're looking at right now, actually.
Okay, good. Okay, so that peptidoglycan layer is like a pretty tight net. It's kind of hard to get stuff through there, but it's also like if you get DNA through that first membrane, let's say
Yeah.
you're phaging, you eject through, you have a spike that injects the DNA, you're then stuck in that periplasmic space between these two membranes. There's a protein called ton B, which is typically used to transport iron or vitamin B12 across the membranes, and again, ATP dependent, so it requires energy to do this. We think the phage DNA somehow gets transported across by ton B. Again, don't know how. We know it can be a transporter of things across the membrane. We know knocking it out can engender resistance to some phage, so
Yeah.
it obviously it's phage specific. But the phage world has a problem where it's like, yeah, we know these receptors are important. We don't know what the phage and the bacteria are doing. We don't know what they recognize. We don't know what
Yeah.
changes. We don't know why some phage have no effect on hosts and some have have a
Yeah.
huge host range. Some have very specific host ranges. We haven't got to that level of understanding yet, which
Is
is
it
kind of crazy.
is it so when we think about like biology, or at least when I think about biology, there's this
Yeah.
huge what's the right word? I don't want to use the word design, but biological design space
Yeah.
where, and especially with something like phages, which I don't know anything about phages except that they are the most abundant biological
Yes. Yeah.
things,
Thing.
life forms,
Yeah.
things on the entire planet. And if evolution directs, in my mind, if there's a
Yeah.
clump of proteins that needs to get inside of a cell, if it happens to bump into whatever surface protein a little bit better than another one, evolution will take that and roll with it. So in my mind, or my question, I guess, is it as simple yet complex as that, where evolution took a path that doesn't necessarily make it easy for us to understand why, but it's utilizing a surface protein that maybe no other phage back, a billion years ago was utilizing at that time or whatever. Do you kind of get what my question is?
Yeah, like it's kind of like a shotgun. Like the simplest way in is just whatever works, right?
It,
That's what
eggs.
evolution
Yes.
works on, like that scale where if it works, it doesn't matter if it works badly as long as it works better than everything else.
Yeah. Yeah. It's
Yeah.
just taking
Um.
advantage of something that's already there, especially if
Oh yeah,
nothing
yeah.
else
So
was taking advantage
the
of it.
the receptors obviously on the phage on the bacterial surface are not phage receptors, right?
Yeah.
They're doing stuff with the bacteria. So the phage and bacteria again in this kind of co-evolution game all the time. Like it needs that receptor for some environment. If it didn't, evolution probably would have killed it a little
Yeah.
while, killed that receptor at some point. So it's just kind of adaptationist, but there you go.
Yeah.
It has a value for the receptors have some value in some environment.
Absolutely. Um,
So yeah, the potential diversity in phage biology as well again is probably the most in the natural world, just from the age of the phage and bacteria interactions and how many phage there are, right? So if
yes.
you've got 10 to the what was it 10 to the 14 phage or something, some ridiculous number. It's like an order of magnitude more than everything else, and they've been there for probably as long as bacteria have been there. So this game has been going on for two something billion years probably.
Yeah. Would you
Yeah.
so I'm now back on the slideshow. You can kind of see which
Okay.
one I'm hovering on. Oh, are you
Yeah.
taking a peek at some other things in the future? I see your
Oh no, that's on my other screen. I'll get rid of that.
okay. No, no, you totally can. You absolutely
No, no,
can.
yeah.
It's just
So
you're on the slide that I was most interested to like take a closer look at after I
yeah, yeah.
found it.
You gotta look at that one.
Um, would you mind just we sort of like talked around it,
Yeah,
but
sure.
how would you define a virus? How would you define a phage?
Yeah.
And I'm gonna ask the question everyone asks, which is,
Good
are they
question.
alive?
Are they live? So a virus is anything that is a uh, obligate parasite. So it has to parasitize a living cell. It,
Yes.
it it can't do its own metabolism. It can't reproduce its own genetic code, be that DNA or RNA or double stranded RNA. It can't make its own code. Um, it used to be considered like it had to like reduce the fitness of the host, but I think that hasn't that's not true anymore.
Interesting.
We have
That's
some
yeah.
yeah like because of parasite to be a parasite you had to do you're either a mutualist or a parasite or there's a third one somewhere in the back of my head
Alt Miss Frizzle will
but
know.
yeah oh, so like it but we I think that description is kind of old fashioned now so
And can I quickly ask you then,
yeah
so you work in bacteria, but
yeah
obviously also, you're an expert in viruses, I would say, or phages at least. Are you aware of the theory of the evolution of introns in eukaryotes?
yes
that they are like ancient
So
viruses or
yeah,
ancient genomic invaders, maybe is like,
there's a fantastic one which I love to talk about, which is what's its name? Whatever its name is, it's involved in the formation of placentas. So
yes.
when mammals, yeah,
I
you
know what
know
you're
the one I
talking
mean,
about.
yeah,
I know what you're talking
yeah.
about.
Oh, my
Your
cat. Sorry,
camera
this is
just
the thing.
died.
My camera. There we go. Let's turn that back on. See if that works. There we go. When you want to make a placenta in a mammal, the outside surface of the placentum organ is one single layer, and the way it tells all those different cells to make the single layer so it's impervious to small molecules, viruses, bacteria. Basically, it seals off the fetus from the mother and allows nutrient transmission, all sorts of things. But it's a single cell layer which you don't see anywhere else. Placentas are incredibly complex organs.
Yeah.
That process is derived from a viral retrovirus, which viruses often need to tell cells to move out the way and open up and change their cell structure. So it's the same code that effectively Ebola uses to tell cells to split apart. It's the same. It comes from that same lineage of genes somewhere in ancient history, at least 60 million years ago. And we use it. Mammals use it to make placentas, whereas viruses use it to cause massive blood loss.
Yeah. And this is not the first time, certainly in evolution or even the evolution of humans, that this has happened.
Yeah.
So I know we have like an ancient degenerate HIV type sequence found in our DNA. The thing for chat, what I mentioned with introns, which I'm not exactly going to explain, but they're an important, essential gene feature of all eukaryotes, whether you're a fungus, you're a single-celled guy, you're a tree, you're humans. There's a hypothesis that these sequences, which are ultimately taken out prior to being used for coding for a protein, originated as a DNA invader. And sort of the reason for that is because we can find DNA invaders that look very similar to the machinery that does it. And it does the same mechanism of picking them out of the genome and moving them somewhere else. Our introns don't do that. So there's this idea that we've co-evolved with viruses. So that's why I'm surprised that I had never really thought that there would even ever be a definition where a virus has to be detrimental to the survival of an organism, because
I think it's a logical chain where it's like it's a parasite,
yeah,
and parasites are defined, or at least used to be defined,
I mean
as
pre the
reducing the fitness.
pre the genomic era or genomic era. I'm sure
I think it's useful to think again though the outside cases, the ones that don't conform to that, are really interesting.
Yeah.
And probably common, but it's a lot of our biological knowledge is based on what makes us sick.
Yeah.
And it's
Yeah.
when we care about what is a pathogenic virus doing. Did you ever watch the BBC show Red Dwarf?
No, I haven't actually. That's on my list of sci-fi
Yeah.
spots that I need to go back and fill in.
They have good viruses in, I think, one of the latest seasons, and the last proper season they had these good luck viruses where if you get infected with this virus and then you just have unreasonably good luck for like 10 minutes before your immune system like works out there's a virus. Or the sex appeal virus. And it's like these traits we consider inherent to some people are actually just because they're sick,
Yeah.
or infected, or
I mean, the
it's
luck.
very kind of like,
Let's talk about that. I mean, the luck. I don't think you can genetically encode luck, but I like the idea of a sex appeal virus.
yeah, no,
If you're a
it's
virus that wants to have your host interact with
yeah,
other potential hosts, what do? You mess up their pheromones if their species uses pheromones, or maybe the pheromone receptors, so that they just get a little bit more frisky and want to go meet others. Suddenly, you're moving along to a new host.
there's
That's
some work with, it was either chlamydia or syphilis, where they were showing that people are more sexually what's the word I'm looking for, active
yeah, sure, active.
after being infected with that with one of the, I can't remember, it might be syphilis, it might be chlamydia. Syphilis makes sense because it starts eating your brain, but I want to say chlamydia. I'm sure chat will at some point look it up. But
Yeah, if someone knows what that is.
there was some study, again, it was one of those Don't tell. I just looked at chat, Stephen. Don't tell Trek Rise about this. Yeah. Yeah, it was. I think chlamydia. Anyway, it's yeah,
That's
and it's one of those studies again where it's like,
it's like an association study, but there's no
yeah,
mechanism found behind
yeah,
it. But the idea, like again, this like biological design space, you could logic together a chain of molecular and cellular and finally like whole body events that
Mhm.
may be. And again, evolutionarily speaking, if it increases the fitness of the virus, why not? Why couldn't that be,
yeah,
you know, possible? So yeah, so that's one of the things I like to talk about a lot on these Wednesdays is the why not sort of biology, because again, like science fiction writers, they get it wrong so many times, but at the same time, it's like I kind of like that idea. Maybe we
yeah.
just haven't found this thing.
Mm.
Um, but yeah, okay. So you sort of defined a virus. So then what's a phage? Because obviously you
A
can.
phage, yeah, a phage is just a virus that infects bacteria.
Yeah.
So the ones on the slide are typical bacteriophage. They sort of isodecahedron heads with a neck and then some feet. They come
Very
in
alien. Very just
yeah, yeah,
so just already base level. So strange.
yeah, like machine-like almost,
Yeah.
right? Like you can see. I think a lot of people also think this is what viruses look like mammalian and plant viruses, because they see these images so often. There was I don't know why I was watching it, but I was watching the DVD the commentary for Rick and Morty,
Yeah.
and one of the early, I think season one, they go like the human body part, and they
Ah.
say they get attacked by, as they
Oof.
say, oh no, it's E. coli,
Yeah. Yeah. Yeah.
and it's
I
clearly
remember.
phage. And one of the writers like, oh, we've got in so much trouble with that, and Justin Rowland was like, but how do they know? It was like, because it's clear. It's like saying, oh, that's a bike when it's clearly an airplane.
Yeah.
Anyone who's in the field knows exactly what they are. It's like they know because they know it's the different things. But
They can
there's
tell by the way it is. Like that's
yeah, it looks different.
yeah. Uh, I mean, that's just a fundamental question just for um, like the general public. I remember being at like an outreach event or something like that. We were teaching about CRISPR and DNA because that was and still is the hot thing to talk about because people have heard about it. And I had an elderly gentleman come up and we were talking, and he just asked like the simple question of like, well, how do you know you have the DNA in the tube? I'm like, and like I know the answer, right?
Yeah.
We can measure it with various like fluorescence based or absorption techniques,
Mhm.
dye based things, but at the same time I was like, that's a really good question. We are just moving around blank or clear
Yeah.
tubes of liquid.
Clear tubes. Clear
Um,
tubes.
and yeah, we display. I know you have an association, have had, still have association with Kurzgesagt, which is
Oh yeah, yeah.
amazing, like visuals. But sometimes I wonder, like, do people when they go to the biological stuff, it's so cartoony and fun and visual and engaging. Do people know that that's like we know what these things look like? What
Yes.
we're looking at right now, if it wasn't clear to people, because we didn't even say this is a picture as close to a Polaroid as what you can get for a virus in a cell. Um, I assume this is some sort of, well, it's electron microscopy. I don't know what specific flavor it is, but
I don't know. It's a if it
it's
is, I think it's a composite of several images just from
probably because you can
um
see, well,
yeah.
yeah, because you can kind of see, I don't know if there's what the right term is again for EM, but a Z stack, kind of going
Yeah.
up and down the plane, and obviously
Yeah.
false colored.
Yes.
Unfortunately, things aren't this exciting.
No, it's
Just
all grey. It's all
it's
grey.
always gray. Yeah, but there's this. Yeah, I just feel like there's a little bit of a disconnect. We can say all these things, but I wonder if that was Justin Roiland's more what he meant was like, how
Maybe.
can they know? Like he knew that we couldn't shrink down like Miss Frizzle to go look at things,
Yeah.
but maybe he doesn't know we can shoot electrons and X-rays at things to get an idea or an actual image of what proteins or cells or viruses or phages look like.
Maybe. I think he was pretty adamant that they specifically were
Sure.
not E. coli, because I mean E. coli kind of boring to draw as well. So
Yeah. Um,
It's I once so I used to do a podcast a long time ago with a guy called Miles Power. It was fairly successful. It's actually how I met David.
hmm.
Well, how I began talking more with David Hewlett. Who, for the viewers who weren't here earlier, David Hewlett's the guy from Stargate, Atlantis, and sometimes in the better Stargate, SG-1, which I hate me saying.
Is that a real
We'll talk
opinion?
about no,
Okay.
actually, just like winding him up. The I Stargate SG-1's fantastic as well. But we're gonna.
Okay.
I saw Sci-Fi Girl asked. We're gonna talk about the. She said she saw Phage and SG-A.
Yeah, I
It's
saw that.
on. It's on the list of things to talk about, I think. But so I used to do this podcast and it was about rational thought and skepticism, very very 2000 mid 2000s, and we had the opportunity to talk to an AIDS denialist and interview him. So I don't know how aware you are of UK libel laws, but there was a famous journalist, science writer called Simon Singh. He got sued for libel by the Homeopathic and Chiropractic Association.
Hmm.
Eventually won his case and changed libel law in the UK, which was all great. But we got in some trouble for questioning. I think it was a psychic. We were like basically taking the piss and saying this stuff isn't true. Here's all the evidence. Da, 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 da. And she took offense to us and tried to like copyright strike our videos and podcasts and stuff. And we eventually got it all resolved. But Simon Singh knew of us from Miles. Had been doing a lot of work on showcasing how the AIDS denialism is rampant in parts of the world, very, very dangerous. There was a film that came out called A House of Numbers, like 2006, 2007 time. Very influential film. But Miles made a fantastic, like, almost scene by scene debunking of this film, of this HIV denialist film. So we were on the radar for having done this stuff. And we, Simon Singh managed to get us this interview with this AIDS denialist. And he just wouldn't accept this idea. Like, he was like, oh, but these kind of images, the microscope images, they're fake. Like, because you have to color them and stuff. And how do you know? Like, it all comes out of a computer. And, the computer could just tell you, well, this is what someone's had to program that computer to say, this is what viruses look like. So if you tell it you're going to look at a virus, it's going to show you a picture of a virus. And I was like, I had to take a step back and say, a couple of things. First off, the first electron microscopes actually didn't use a computer. It's literally like a fluorescent pad. You shine the image on it, like a fluoroscope kind of thing. So, not true. Aside from that, when we find these structures, that informs us that knowing the structure of a phage like this isn't that useful unless it provides us with a question we can test, and then we can verify whether that's true or not. So we say, okay, well, if it's this shape, we have to think, what does that mean? How can we now prove it's that shape? What can we design to test that? And HIV has a fantastic thing. I forget the name of the drug now, but when I was more involved in this HIV denialism, it was still in clinical trials, and it's called T20, and it's a nucleotide analog. So basically, it just has. It's a peptide analog. It's a peptide analog, not nucleotide.
Then I don't know that.
So it's basically a little stretch of protein, and all it does, it does an amazing thing. When HIV hits the cell, it has two co-receptors. It binds, was it CRC5 and CXRCR4 or something? It's been so long now. Anyway, it binds these two receptors, and it shoots a harpoon into the membrane. And the way it wrenches itself in, it spins this protein back on itself and up. So it tightens this back up and flicks itself back up. All this drug does is, by knowing the structure of that protein, by knowing exactly what the proteins are, people far more brilliant than I did, designed a little protein analog that will perfectly sit in that space. By knowing exactly what those amino acids are, they were able to design this little protein that will just sit there, and now the virus can't retract its harpoon. So it just sits on the outside of the cell surface, and an immune cell comes along and goes, oh crap, there's a virus on the outside of the cell, and gobbles it up. And then the cell doesn't get infected by HIV. So by knowing these structures in this case, we were able to design a drug that has been proven to improve lives. Yeah. He heard this, was like, oh, but that's all fake.
Oh my gosh.
he was a nutbar.
And
He's the correct
and
term.
Fuvertide.
Yeah,
By
yes,
the
and
way,
Fevertide, yeah.
that's
Yeah.
just looked it up.
It used to be called T20 as it was his preclinical name.
Yeah.
It's an amazing thing.
Huh. Wow, that's super cool. I
Maybe
had
go off the
I had a
slide.
whole
I'm sorry.
question and then I forgot. But no, that's awesome. Yeah, I know. I should have like pre-warned you. Again, I had a very long week, so I didn't get to talk to you offline too much. If we go off on a tangent and talk nothing about what I meant to talk about,
That's fine.
perfect. 100% perfect stream right there. That's what I
Yeah.
want.
As long as
Um,
it's interesting.
yeah, as long as it's
As long as people,
interesting. Exactly.
I
Um,
can talk about Coach as well. I still work with them quite. We have a couple videos coming out, one mid July, and I'm working on another one right now as well. That I shouldn't say what they are,
I shouldn't say
but
what they are.
yes,
Can't reveal them.
I can't. Yeah, I still do work with them. So
Oh,
we're still doing
that's
stuff.
cool.
So
Yeah, no. I'm sure everyone knows what Kurzgesagt is. I mean, it's
yeah.
the millions of views they get on every single one of their videos should be proof enough of that.
So if it's a video about biology, there's a good chance I either wrote it, edited it, or fact checked it. So that's my sort of contributions. It'll be 6 now videos I've written, and most of them I've edited, and a couple I just fact checked.
Wow, it's very impressive.
No, and they are super receptive to actually like Philip, who's the guy in charge. His ethos has always been like, how can we make this as complex as possible but still understandable? Like,
Yeah.
like, how can we make it so simple? It's like these things are complex and they're cool. How do we tell people about this cool stuff?
It's like the opposite. And this is not me trying to derail or re-rail the conversation,
Yeah.
but I do think it's like the opposite of like sci-fi technobabble,
Yeah.
where you take real words and say them as fast as possible so no one catches what you said. Rather, it's the much harder job of slowing down and saying like, okay, where are we starting? What are our terms? What's the space we're working in? And let's move forward so that the people who don't deny the existence of HIV, or I guess AIDS, because I know there's this whole weird thing about like AIDS isn't real, but HIV maybe as a virus is real, but it doesn't cause AIDS. That whole thing.
Yeah. There's also people who don't think the virus is real.
Yeah. I believe it. I believe that totally.
He didn't believe viruses existed.
Absolutely. I mean, I don't think you were on this stream or saw this because I didn't see you then, but we actually read over a paper that studied the genetics of psychic ability. That was like 2 months ago.
Is that Rupert Sheldrake related in any way?
I don't know. I don't follow. There was sort of a last minute like something got canceled, had to do something else. Paper in quotes. It actually they found a repressor of psychic ability and was super disappointing because they didn't actually propose any genes as having psychic ability. Not only was it a bunk paper, but they oversold it in the abstract.
They just said, "Oh, we might have found a repressor."
Well, they found a gene that was negatively, or a SNP variant in people that was negatively associated with psychic ability, and then they proposed that perhaps that was a repressor of psychic ability. It was so uninteresting. It was disappointing.
Was it a GWAS study or?
They did their own.
They just found a SNP.
Well,
okay.
So they did they went about it the way you would do it if you wanted to. First of all, they started presupposing that psychic ability exists and it's familial. That's what they said.
Okay.
And therefore, if it's familial, it's genetic. They administered a test of psychic ability. They actually did some cool things where they asked people who weren't psychics, they had self-reported psychics and people who measured highly on this psychic ability test that they had, and then people who measured very lowly, and also asked, what are your thoughts about religion, psychic ability, that sort of stuff. Then they did some SNP testing on all of them and tried to correlate ability with certain SNPs. Super low N not actually a GWAS. Also, it presupposed psychic ability is a real measurable phenomenon. So
Yeah, and they weren't using whole genome sequencing. They were just
no,
doing like
just simple,
21andMe, like here's
effectively.
like a microarray kind of like, does it bind or doesn't bind?
Exactly. They were using
Yeah,
microarray technology, like
yeah,
not 21andMe, but a similar service.
yeah.
I'm a genomicist, so I was very interested in it until I found out the data were like really boring. So I didn't dig into it myself at all. Yeah, I was really disappointed in that.
I think, yeah. To get back to your other question, you asked me another question. I derailed straight away.
Oh, it's okay. Do it.
The
As
sci-fi,
Altmus Frizzle points out, I derail my own streams constantly, so it's par for the course.
yeah, sci-fi writing is, I don't know. I think there's so many scientists who owe a lot to sci-fi, good and bad. Like, I think a lot of people got interested in science is because of, frankly, terrible sci-fi films and
Oh, yeah.
TV shows, right?
Well,
So,
I don't know your opinion on Jurassic Park, but I'm definitely part of the Jurassic Park generation of scientists.
yeah, it was. I I'd like Jurassic Park. I never thought of it as like, oh, we should be doing this. It was always a case of like, why would they use frogs?
Because they needed something.
Yeah, there was a
Also, have you heard of George Church?
George Church. The name rings a bell.
He's one of the scientists who gets all the money for doing crazy things. He is co-founded two companies, both of which are working to de-extinct
Mammoth.
the woolly mammoth.
Yeah,
Yeah.
yeah, yeah.
Very weird thing. Like if professorship doesn't work out and you want to backtrack, they are actively recruiting postdocs to
Okay,
de-extinct the mammoth.
instinct. I mean, that's cool.
That has like an ecological bent to it that's
Yeah,
interesting and all that.
yeah.
But yeah, Jurassic Park is a couple steps further from that.
There was I just watched a stream earlier today by a science communicator called Simon Clark. He's a physicist, does atmospheric science stuff, but he plays D&D every week or so with a bunch of other science communicators. And they had a one shot today, which was set in a dinosaur themed one shot, and they were asking the chat for suggestions, and they basically ended up picking a couple of mine, which is just, so in the UK there's a town called Swindon, which is like remarkable because it's unremarkable. Like there's nothing happens in Swindon. Like the most interesting fact about Swindon is it has like nice roundabouts or magic roundabouts. That's
Okay.
that's Swindon's claim to fame, a roundabout. And it just derailed into absolute hilarity with like this UK crappy science Jurassic Park knockoff. If anyone, I'd go watch it. It was
Yes.
they call themselves
Simon Clark, my mod Brian. Maybe I can call upon to look him up and put the channel in the chat. Simon Clark. I wrote it down because I wanted to look it up later.
Dr. Simon Clark is his Twitch channel.
Cool.
And what do they call themselves when they do this Dungeons and Dragons thing? RP Geeks. That's it.
RP Geeks. That's a good name.
Yeah.
Yeah. Also, Genome, please don't bring back dinosaurs. We can't handle it. LOL. No, we've definitely proved that as a species, we can't handle a lot of things. But
the classic joke has been, watching Jurassic Park. It's like, why would people keep building this park? And after the pandemic, it's like, oh yeah, no, that makes sense.
It makes a little bit more
people
sense.
100% would just keep going.
Yeah, and the incentives for it also makes a little bit more sense now that I've been off stream playing a little bit of Jurassic World Evolution 2, Jurassic Park building game. You just get so much more money and attention for having big, dangerous dinosaurs. So I keep filling my park full of the worst dinosaurs, or the most dangerous dinosaurs possible. Oh, cool. Thank you, Brian, for those links. Yeah, I'm actually, if you know any paleontologists, I've been looking for someone to come talk to me about Jurassic Park because I want to talk about the genetic engineering side of it.
Guy paleontologizing. He has a Twitch stream.
I think I know it. He's pretty big, isn't he?
Yeah, he's kind of big. I think David and him have talked a couple times, so there might be a way there. Um,
It's not.
he streams. Yeah,
It's not essential.
yeah.
But I would
But,
love to talk to somebody like that. Yeah. Um. So I want to take you to the most boring slide I have on here,
yeah.
which is this thing called Aerosmith. Have you ever
Yes.
heard of Aerosmith, like the book?
The book.
Yeah.
The
Okay.
book. Yeah.
Okay.
Yeah.
Yeah. So I only just learned of this today.
Yeah.
It's also a book. No one's ever made a movie or TV show of it, as
No,
far
there
as
has
I.
been a movie.
Oh, there is a movie.
Has been a
Okay.
movie. Yeah, there's a movie. There's a movie. Irish.
Well,
Yeah.
um, for everyone else, because I failed to find a movie image of it, it is a book from, I have it here, 1925, that talks
Yeah.
about using phages to fight disease. Now, isn't that interesting? That's,
I mean, it was a pull. I think it won the Pulitzer Prize. It won some award.
yeah,
when
I
it
saw
came
that.
out. What's the author?
Sinclair Lewis.
Sinclair.
Yeah.
Sinclair. Yeah. He based it on a guy called Durrell, the French, one of the discoverers of phage. He discovered it. Him and Twort discovered phage independently of each other. Durrell slightly later, but Durrell did a bunch of the early work. Thought basically said, "Oh, there's these things that eat bacteria. Isn't that cool?" Then when fought in the First World War, Durrell was like, "These things are really useful. Maybe we should do stuff with them." They didn't, and it's completely independent of each other. So, Durrell was the first scientist to kind of really look up. Yeah, there you go. There it is.
There it is. I didn't know it was
Yeah.
a movie.
Yeah.
A very old movie.
Oh yeah, very
1931.
So Durrell, the character Aerosmith is kind of based on Durrell, in a lot of ways. Durrell is a really, really smart guy. There's a lot of people who've worked on phage are like kind of pseudoscientists because it's like, okay, well, it was a back end field for a long time. People, it was done in Russia. Even today, there's lots of papers that like have been translated from Russian, and it's like, well, hang on, sorry, you did what? Like, you're not telling me what phage you used, how much you used, what way you applied, like lots of bad science.
Yeah.
Although,
can I ask, are those from like pre 1980? Because a lot of those, a lot of those old papers in English are pretty loose on details.
Terrible. Yeah, like three references and it's nature. Yeah. There's been a lot of poor work and Durrell was not one of these people, and I just assumed he was because of some of the affiliations he had later. And so I was like, okay, he just was the second guy to discover phage. Good for him. And then I read his work, and it's from the early 1920s, and 30s, and it's like this guy was one of the best microbiologists who ever lived, and it's a shame he's not better known for some of the stuff he was suggesting and thinking and doing. Like there is the topic, like the thing I suggested, using phage as a means to reduce antibiotic resistance and virulence. So here's the other thing. So we didn't mention, not just phage don't just bind to the efflux pumps. Some of them bind to virulence factors. So the things that bacteria use to make us sick. So when we add these phage, the bacteria that develop resistance now are less virulent. So now the patient doesn't die. Even though they still have a bacterial infection, the bacteria isn't actually killing them. And Durrell had that idea in 1920. It's like it was just hidden in an old French-Russian journal somewhere. It's like, and he clearly stated it, and it worked. And he showed that people who got cholera then didn't transmit a virulent form of cholera to other people, even though they were still producing Vibrio cholera type. It wasn't called. I don't think it was called cholera at that point, but they were still infected. They just weren't transmitting it to other people. And again, all there. So, Aerosmith is based on him in some sense. The actual character Aerosmith is kind of more of a dick than I think Durrell ever was. But so it's another fun. So go ahead.
Well, I was just going to say, so this came out, the movie was 1931. The book was in 1925. I'm just trying to remember my scientific history. I don't even think I'm trying to think when Thomas Hunt Morgan even proved that genes were carried on chromosomes. This was I think it was a little earlier than this, so that might have been out. But this is, I mean, to put things in perspective, I think for the audience, we didn't always know when Gregor Mendel discovered genetics, he didn't know how it worked. When Darwin came up with evolution, he didn't know how traits were carried really either. We had some wacky ideas about it. We literally didn't know the molecule that carried it. The thing that oh, your camera died again.
That's right.
Uh,
Yeah, it keeps.
it's okay.
I'll fix it at some point. This is the first since I had a problem earlier. This is the first time I started doing this.
Oh, interesting. What was I saying? In terms of like the history of science, like these ideas were coming out right during like the modern sort of synthesis of sort of modern science. There was this shift in I think the 1920s and 30s in biology, where it was becoming less old white man in a tweed suit, like poking around in nature, and more, okay, this is an experimental science. We can make theories, hypotheses, we can do experiments and test things. The idea that the ideas we're working with now with modern molecular biology were all back all the way prior to any of my grandparents being born is insane to me. Morgan published the chromosome paper in 1910. Okay, so it was a little bit contemporary.
That's still
But that's still only like, we didn't know like that Drosophila and a phage necessarily even were carrying the same molecules.
Well, there's DNA, the DNA being genetic materials, Avery, which I think is 44 if I remember, like showing that you could that it was DNA that was covering
specifically. Yeah.
Yeah.
I guess you're right, because chromosomes, like I'm sure they were doing all that sort of old school biochemistry, protein. And they thought, protein. Yeah, exactly.
So Erwin Schrödinger's book, What is Life? I think is 43 or 42.
Yeah.
And that, excellent book. Everyone wants to read a really great old, what if science book. Erwin Schrödinger's What is Life is a magnificent read. And he kind of lays out this argument about why it's protein, and it's wrong. But it's like, it's all testable. So yeah.
It's just that they couldn't test it at the time.
It wasn't easy. Yeah.
Also,
even Avery stuff got pushed back tons.
To go back just a tiny bit, because I don't think we even said this when you were talking about these crazy little microscopic creatures, or whatever you want to call them. They are just, what we're seeing is just protein, except for this little bit coming out, which might be DNA, depending on what they're actually showing. There's nothing else to these. I mean, it is DNA with a fancy protein shell. Cells are not much better, but quite a bit more complicated.
Well, cells also. So you asked me the question. I completely skipped it because I was too excited. You asked me, are they alive?
Yeah.
Right. And I think one of the defining features of life is ATP.
Sure. And
how cells regulate that homeostasis between energy production. That to me is the defining thing about life. So I actually don't think viruses are alive.
What about virus?
Yeah, it's in that weird position, right, where it's like, oh, maybe it might be alive.
Yeah.
I think life is a weird distinction to try and pin down.
Absolutely. I think the question itself is flawed. It's just the question everyone always asks. I don't have the right words to describe it, but in my mind, if you have a transposable element that is just a string of DNA that only lives inside of a nucleus, that only encodes for the proteins that help it hop around, in my mind, that's a form of what we might call life. It's made of this class of molecules we have agreed are biologically based. It lives in and within what we definitely know as life, and it evolves. I mean, but you're absolutely right. The homeostasis, the energy metabolism, that's all missing. So it's not the same.
It's not the same.
But I am a biology liberal or something like that. Everything's alive.
Healthy. So here's a question for you then. Mitochondria. If I take a mitochondria outside of a cell, is it alive?
Is it alive?
Yeah.
Alive, no. In the same way, I still would say that that transposable element outside of its environment
Yeah.
is not alive. It was alive. Just
Yes.
like you
Yeah.
take me and take me out of my
So
biosphere and throw
yeah.
me into space. I'm not alive anymore.
So you said it was alive.
Yes.
I assume you meant when it was inside the cell. What about 2 billion years ago when it was a free floating bacteria?
Then it was alive. I definitely think that
Exactly.
I definitely
Yeah.
think
Yeah.
that the
I agree.
characteristics
Yeah.
of life can be gained and lost. In fact,
Yeah.
so this is another thing I wanted to sort of ask you about, but I wasn't sure your expertise on this. It's certainly better than mine. Is the top down or bottom up evolution of viruses? Did they evolve from the
Yeah.
same random bits of DNA or RNA molecules and gain some, but not all of the characteristics of cellular life? Or are they cells that have lost
Yeah.
components of
Given
themselves?
I'm of the opinion, given the diversity, like double-stranded RNA and single-stranded RNA, the different, like basically any way of making DNA has or RNA has become a virus. I think that's bottomed down. Basically, like these bits early on
Yeah.
began paralyzing, like attacking other things and taking over. I think. And that's just not to say that again that it could be both sides. There could have been viruses that jumped out. I mean,
That's my
that happens all the time.
yeah, that was sort of my opinion. I think because
Yeah.
I think some of those big viruses. So I mentioned
Yeah.
for the chat, Mimi virus, which at one
Yeah.
time was the biggest known virus. It has all sorts of cellular components that you don't expect for a virus, and I thought that there was a good evidence that it did evolve from sort of a degeneracy
Yeah.
in air quotes, because what does that mean in evolution? It
Yeah.
evolved from what we would call cellular life, because it was there's certainly endoparasites that are cells in other cells, and perhaps evolution took it down a road where it just lost some of its free living cellular components.
This is why I brought up mitochondria, because I think it's a great distraction when people ask me this question. It's like
Yeah.
through evolutionary time, most people think of evolution as like getting fitter, like, and that that implies living more, breeding more, doing those things, and being alive. You'd like to evolve, you must be alive. It's like, well, no, mitochondria evolved to be dead, and that was the
Yeah.
most successful way
Yeah.
for them to replicate, was to be dead inside a living. It's our definition. Our thing of like, well, this is life, this is not life. Here's a nice, neat line.
No.
Is hard. I mean, maybe there is a line and we just don't know it. Like I don't know how much stats you do, but like there's a thing called SVM modeling, like support vector machine learning. You try and take a hyperplane and draw a single plane through it. And there's always a plane that separates the max, and it's just mathematically shown. There'll always be at least one plane that will separate these things out maximally.
I see.
In any dimension. And I'm always like, well, that's is that not true?
So you're saying if we can measure all n dimensions of every characteristic of life,
yeah, would we find a plane?
it's got to be. That's very interesting. I actually haven't heard of that. I'm gonna use that, even though I am of the opinion that it's such a gray area that.
Yeah.
But you're right. I mean, mathematically, it makes sense that at some point that gray becomes black or white or whatever. That's interesting. I like that.
But yeah, it's sort of like you could have a bit of A, a bit of B. If you have A and C, you're alive. You have B and C, you're alive. Then it's like, well, that doesn't work anymore. Like they have to be discrete blocks from each other. But either way, it's always like I think as you said, the question is kind of flawed at our current understanding.
Yeah, absolutely. And I to reiterate your own point, like I have always thought some of the most successful organisms on the planet now, in terms of their civilization and technology, are the DNA invaders that have lived with us since we were just learning how to walk. Like, what effort have they put in? Nothing. Yet they're just along for the ride, and they are unequivocally, and I'm not even talking about the ones that even replicate themselves anymore, the ones that are long dead. We still have those in our genomes from millions of years ago. Crazy. It's fascinating. Whew. Good question. Good questions though. I could talk about definitions all day long. the last chapter of my thesis was basically me arguing to the air about how we define the certain things that I studied as a graduate student.
Yeah. No, it's philosophy is just arguing with dead people.
Yeah, that's true. All right, so let's pick another just topic.
Sure, sure.
Just I don't know, sort of
I,
randomly.
I,
So I
yeah,
I know some people who followed David's stream. I think
yeah.
there might still be some stragglers. Some people might have finally dropped off after several hours. I mean, I was on David's stream like at 2 p.m. today.
Yes, he started he
Oh my
started
gosh.
3-ish and then
Uh,
carried on.
so we can talk about Stargate. This is one that's sort of like pretty cut and dry. I don't.
Hmm.
Oh yeah. So actually, what is your relationship with science fiction? I didn't really ask. So mine started with just as a child being exposed to Jurassic Park, and as an early teenager, Stargate, Star Trek, that sort of thing. But what about you? Did that have an
Watched a lot
effect
of.
on your
Yeah, I don't.
trajectory?
I don't know. Like I watched a lot of Star Trek and Stargate when I like at university. I watched like the. I was saying on my stream earlier, like, I would watch Stargate Atlantis when I got home from university every day because it was on TV, and we enjoyed it. I didn't. I never assumed I'd be playing D&D with a guy at
Yeah.
some point. But I watched a lot of like Star Trek, and I was more a fan of Star Wars. So uh,
Oh.
that's less sci-fi based.
It's science
But again,
fantasy. Yeah.
yeah, um, I I enjoyed it. I don't know how much the philosophy of Star Trek played on my like scientific thing. The biggest impact on my scientific like journey as a child was probably Miss Frizz, like Magic
Yes,
School Bus.
Miss
This is like as a
Frizzle.
young kid, yeah.
Yeah.
I saw one of the people in chat had the alt Miss Frizzle,
Alt
like
Miss
yeah,
Frizzle.
like yeah.
Which I know some people came in after that, and I don't think I think she's listening. Hold on. She has just begun streaming herself.
Okay,
Uh,
but so
anyways.
yeah, the Magic School Bus was definitely like from a very very young age. That
Yeah.
was like, oh, this exploring the world. There was also an old TV cartoon. It was big in Europe. I forget what his name was one now, but like they had different seasons where they did like one about history, one about science, one about the body, and it was I think it was originally Spanish because my Spanish friend was playing it to her kid. It's like I used to watch this as a kid. And it was all I found out that it was actually I think it was actually Spanish at that point. Um, but the like TV sci-fi that really got to me.
It could be a book
There was
too.
yeah, well,
I'm very
so
visual when it comes to like the
yeah,
media that I consume, but it could have been a book.
the horrible histories books. They did horrible science as well. I don't know if
Okay.
you like again a
I
BBC
haven't heard of
TV
that.
show in the late 2000s, but when I was a kid, it was a book, and these were like really fun, like dense, like 2 or 3 pages about a single thing, and then next 3 pages about something else, and it was like, oh, the Black Death was spread by a bacteria that hopped on fleas and bit you, and then and then you have horrible pussy bowl, and it was very very gruesome. It was kind of like horrible science, so everything had to be kind of like tell it in a way that a 7-year-old boy would find fascinating, and they had a great thing. Those books, again, I actually bought them for my nephew because it's like these are the books that single-handedly ignited a lifelong passion in science.
Yeah.
More so than any TV show. Again, it's always cool to see like, oh, science and TV. As a kid, I'd always relate to the scientist on the show.
Yeah.
But yeah, it was definitely those books were the thing that informed me of science more than TV. But
Yeah,
I
absolutely.
enjoy science fiction.
Yeah, no, no, no. I mean,
So,
I shouldn't even say like
yeah,
science fiction is like a sort of is like fuel to the fire, but that
yeah,
fire was started. I have such a clear, distinct memory of being like a 3 or 4 year old, couldn't
yeah,
sleep during daycare, and my daycare ladies, uh, high school age son, who is now a science teacher, used to keep insects, and he would
yeah.
take me and show them to me, and it was just like, all right, The neurons have now connected. Off I go.
It's done.
Yeah.
There goes another scientist in the world. Yeah, no, it's I I think I was also just always a nerdy child. So again, it's like
Yeah, yeah, yeah.
self-fulfilling. Probably like people are like, oh, he's a nerd. Buy him science stuff.
Yeah, no,
Like,
there's certain proclivities that turn us into scientists over
yeah,
time. Yeah. So you've seen obviously Stargate, Star Trek. Okay, so
yeah.
that's good. Have you seen X Files?
I used to love X Files.
Dana Scully.
Yes.
I did not have gendered role models really as a child. I don't know why. I guess I was just weirdly progressive as a child. Oh, you know, it was the red hair. I know you can't quite tell, but I have red hair.
Oh,
Uh,
okay. So it was
I was like a
identifying like, yeah,
red-haired scientist person. I
yeah,
gotta be like this person. Love the X
yeah.
Files.
They did books for kids at one point, X Files, which I didn't realize they were kids books, but as an adult, I was like, oh yeah, no, they used to have like X File, like the X File stories. Like I read them as a kid, and I was like, no way you read like that. It turned out that they just churned the TV show into like a Pulp Fiction
Oh,
book for kids. I read all those as a kid, and yeah, again they very much a positive influence of like, okay, well, rational thinking. What is the actual? But also like, oh, but is it aliens? Is it?
yeah, yeah.
So it's, again,
I just hated Mulder so much. He just wouldn't listen to Scully. She was so smart and so right.
yeah,
But well, that's
yeah,
the weird backhanded like '90s weird thing where it's like she's smart, intelligent, powerful, but she's still wrong because she
yeah,
has to be because it's the '90s.
yeah,
Yeah. Anyways,
So I think a show that did my favorite sci-fi show is probably Fringe.
love
Um,
it's the X Files of the late 2000s.
yeah,
Love Fringe.
exactly. Yeah, it's it's perfectly done in the fact it doesn't like the spoilers for chat and stuff. They haven't seen it, but basically every problem in the show is caused by one of the main characters without him really knowing it,
Yeah.
and it's all based on the work he was doing and how he was trying to help his wife and son in effect. But it's great.
Did you know that so X Files and Fringe have very similar
Yeah,
DNA? There was another show that was never made that was going to also be in the same sort of vein. It
yeah.
was a show that was going to be called CRISPR, starring Jennifer
Okay.
Lopez.
Oh,
She
I
was
didn't
going
know
to
that.
be an investigator of biological like crimes and mysteries.
Yeah,
It was never made.
there was
Unfortunately.
unfortunately there was another show. I forget what it was. It was around that time, and the show starts with like a outbreak of a pathogen released from a lab. Um, and
Helix?
it maybe Helix. I think it was Helix. It had like I remember the opening title having quite a nice CGI thing.
Yeah. That just
I
resurfaced
forget.
for me on Netflix.
Oh, I
Weirdly.
was on Netflix. I have to watch that. Like it was, yeah. Someone in the chat said, said Bones. Bones is also a great. Here's a case where she's almost like I don't know if you've seen Bones.
I haven't. I need to.
It's good. I wouldn't call it sci-fi so much more as
Sure.
like procedural using science to
Yeah.
solve questions. But yeah, the main character Bones is quite often right about things, and it's very much moved on from that '90s where you can be X, Y, and Z, but you can't if you're those things you have to be wrong because the plot requires you like aliens to exist, and it's a great show though.
Hmm.
I think Helix sounds familiar. Someone else in chat said Helix.
Helix
That
is an
sounds that sounds right.
older. It's like 5 to 7 years old. I think it had 1 or 2 seasons. I
Oh
remember binging it. Oh, maybe unless they made new stuff and I just never saw it. I
no, I was
remember.
thinking that it was when I was doing it was before my PhD, so it's got to be
Oh,
the
maybe
thing I'm
older.
thinking of is like a decade old. Yeah,
Let me look it
it
up.
would have been around 2006, 7.
Oh yeah, no, 2014 is Helix.
Okay, so it was a show. It was a show prior to that, then I think. Unless at this point, time is time is
Irrelevant.
blown out the window. It's like
Yeah.
sometime before the pandemic.
Sure.
Like, in the olden
Yeah,
days, there was
sure.
TV shows.
A lot of stuff happened sometime before then, but who can say when? Um,
I know David knew someone on that show, so I'll have to ask him because he was like, "Oh, I can get the CGI guy who did that to do something else." Like, I'll have to ask him who it was
Oh, cool.
because he was involved with that. Yeah.
I need to.
Um,
I need to uh, politely respond a second time to an email I sent to David Hewlett. He responded to the first email I sent
yeah.
to him because he happened to be reading the same book I was reading.
Okay. Yeah.
Uh, Shoot, what is it called? I'm forgetting what it was called. And I was actually thinking about discussing it on stream. I was like, wait a
The
minute.
biotech book, right?
The biotech book. I'm forgetting what it's called. It's got
He
some
told me
generic name like the genetic revolution
Revolution.
or something. Yeah, it's not that, but I'm forgetting what it's called.
David's great. I did send him a message saying if if he I asked him if he wanted to come on. He's too busy. He's streaming all day. He's like, you might
Yeah,
enjoy this chat. He's
yeah. No, I,
he's great.
I
If you
understand the like sort of he is a public figure, so and I'm just
Yeah,
little old me. I sent him an email once. He responded positively.
yeah.
Just give him a couple more days, and then I'll email
Yeah,
him
no,
again.
no, no. He's he yeah. I'm sure he'd love to come on because he loves talking geeky stuff all the time.
I've noticed. That's why I'm a still a huge fan. Uh,
It's bizarre, isn't it? Like one of the guys who played horrible, as in personally horrible scientist, who has a very good redemption arc in the TV show, turns out to be such a nice person who also really likes
well,
technology.
like I mean,
Ooh,
so I again, like his portrayal of Rodney McKay, I would say was also an inspiration for me to obtain my PhD, and it was because I was old enough at the time to recognize like that he is a complex person. And he
yes,
is a complex person with complexes,
yeah,
and but underneath it all was a genius, which I do not have, but a certain amount of I don't even know what the right word is. The problem solving, the portrayal
yeah,
of solving a problem was such a like a black hole sucking me in. Like I wanted to be the person who could solve problems, and
yeah.
now I run around putting out
Solving
fires, solving problems all the time in lab.
life of a postdoc.
Yeah, exactly.
So this is where I get to transition to be a PI. Like I'm just going to cause problems now. Let people fix it for me.
There's gonna
That's what we do, right?
there's this thing where it's like there's a day when everyone for the last time without realizing it stopped going
Yeah,
outside to play with their friends. There's going to be a day in the future where you run your last PCR. How long
yeah.
is it going to take for you to realize you stopped going
That was
into
awesome.
lab?
Yeah,
Sad day or
yeah.
happy day? I don't know. It depends on how much
I
PCR you've done.
I think it's sad. There's a PI in my building who is full professor, tenured, tons of papers, tons of grant money. He still goes to the lab because he's like, that's what I like doing. Like I find the time to do it. And there's his, who's actually my old PhD supervisor is now in the same building. He's like, he never came to the lab when I was my PhD's never been in it ever. And he's what, 10 years older than I am. So it's like, okay, I've known you longer than when I started my PhD. The entire time you never once went to the lab. I'm at the same age you were when you stopped going to the lab. He's like,
Yeah.
well, um,
Genome hopefully is encouraging both of us. Don't
yeah.
stop. Keep doing the experiments. I hope to. I love experiments. And also,
They also released Burkholderia pseudomallii. Oof.
I don't
So it's
recognize that.
a really nasty lung pathogen.
Ooh,
Um, we get
yikes.
it a couple times when we do. So I work with CF patients quite a bit, and if we see that on plates, it's a disaster because it's for a number of reasons. It's nasty, really, really bad.
And this
Have
is a
you
good.
how plate buttons work?
Have you not been at an institution where a professor? Okay. At my grad school institution, a
Feel your pain.
professor announced to the entire department that he was leaving the department for a new job by replying all to an email from like 8 months prior that was sent to the entire department. Like it was like for an announcement for a symposium. He replied all and was like, "Unfortunately, I will not be able to attend this as I will be in Cincinnati at that time because I am taking a job." It's like,
Yeah,
oh my gosh, yeah,
we have probably once or twice a week we have, and I don't want to sound mean or anything, but like an administrative person will get a qualification or certificate, or something like what the recent was. So and so has just become a finance regulator level 2. Email to the entire department. It's like, don't do it. Don't do it. 3 seconds later, reply all. Congratulations. Da, da, da. I was like, oh, they're gonna start. Like, luckily, I found out how to mute those emails like
Oh.
recently. So there is a way to mute them. On I was like, no, because it's just. I see people who are like, you've got more important things to be doing than replying to this email. Yes, it's great for it, but like I just had a paper published. I didn't get like this is why we're all here for science stuff. I just got like $200,000 grant funded.
Ooh.
Like, why am I getting?
Well, congratulations.
Like, that was. I didn't just. It was like I'm saying. Like once I've had 2 R21s, and it's like the level of success there compared to this administration person who can now use Workday a little better seems very very disjointed, and it's like, well, just don't stop replying. Please stop replying more.
We've had such is academia.
Yeah. That'd be fair. That's the worst of our troubles. It's like, yeah, it's a pretty good life.
Yeah. Well, hopefully, I'll be on the job market. I'm skipping I'm skipping this better. November. I'm skipping.
It's gone better.
I've heard.
I had an offer last year, which I turned down. And everyone's like, you're nuts. I was like, I don't know. It was in Oklahoma, and it was
interesting, not
great.
Yeah.
The people in the department were nice, but it was like it was not a step I wanted to make to that town. And they interviewed my wife for a position, and they're like, nah, we're not sure. It's like, okay, well, that makes me a lot more comfortable saying no straight away. If you're not sure, we're done.
Yeah.
So we turned that down. And then nothing. Job fucking crashed. So yeah, it was fun being in a postdoc for a little longer.
So I have a sci-fi question for you then.
Yeah.
Do you remember this episode?
Does
it look familiar? There's 2 screenshots.
Yeah, this is the one on the right is Jules Satir. How do you say her last name? Jewel. She also was in Firefly. And this is she's getting turned into a Wraith hive ship, if I remember.
So this is something that some people in chat might recognize because I talked with another mod of mine all about living spaceships at this point, like 2 months ago.
Yeah.
And we talked about this. Do you remember how they solved this problem? How in a single episode...
This is the phage.
This is the phage. Yeah, exactly. How... So first of all, let's give them credit. A real application, probably, of a real-ish technology, right? I don't know how common we're thinking, sort of phage therapeutics are or will be in the future, but it's within the realm of possibility, clearly, right? Mm-hmm. From what you have described already. Yeah. The question from you, I guess, is like, what would you need to get this out? You were presented with the screen on the left and a patient on the right.
Yeah.
What goes into how? How? I don't have a good question. So, how?
Yeah, if the thing on the left is like a bacteria, like it has to be a bacteria.
Maybe I don't know what that's supposed to be showing us, but sure, yeah.
Like on the left thing, it looks like a compound structure, to lack of a better word. Sure. Like it looks like those circles. Depends how close we are to that location on it as well. Like if we're weighing up, those could be like protein capsules.
Let's just say they're like it's surface proteins or something like that.
Yeah, that looks more like a virus.
They just call it a wraith pathogen. It's in a different galaxy. It's fine. We can sort of sweep that under the rug.
So you need to find something that either binds to it. So again, assuming that they have the same biochemistry, same amino acids and everything, it wouldn't be crazy to find, okay, well, we'll just, what I would actually do is say, okay, what is the antigen that would target this? Yeah. Because antigen library, we know you can always make an antigen to any protein. Like it doesn't use it you could develop that way.
Can I ask, So also for people in the chat, because I'm going to ask this question, I hope it's not too technical. Um, uh, with phages or antibodies or any
Yeah.
two proteins that recognize each other, it's all about fitting. Proteins are just squishy Legos that fit together. That's all they are. We can make random or semi-random bits of DNA that encode for different proteins, and then we can select for ones that maybe bind other things
Yeah.
better. Or have we gotten to the point where we are making phage recognition libraries? Can you do we know enough to randomize the bit of the phage genome and just throw it at something, whatever your new thing is, until you find something that literally sticks?
It's called phage display.
Yes.
So it's I don't know if they do it randomly or how wide of a gamut they look when they screen, but it's effectively a tool that they use upturn phages to try and find stuff that binds. And it's on the tips they put let's say they put random peptide structures and say what when we flow something over this, what got stuck and which point in the display was it.
Hmm.
So it could be done.
Okay. Is that how you would go about that, or are you thinking more direct?
I think I just use fire at this point, but if I had to,
Actually, I think they showed it was like fire resistant or something weird like that.
yeah,
Like it absorbed the energy. I might
yeah,
be mixing up different even properties, but there was something
it
about it. Also,
yeah.
they can't kill off main characters too much.
I mean, they killed off Carlson and then brought him back as a
Well,
clone.
yeah,
Um, I think yeah, it's you could use that technology to say, okay, well, let's find something that binds to it, but then you have to the hard part, hard part of this is, so let's say you have a phage tail that
sure.
binds. How do you then transition that to, well, somebody has to either just break it. You could use like a Taylor sin, which is how there. I'll run through the first. You could use a Taylor sin to break it open, or like a normal phage where you inject DNA or genetic information to it, which replicates and spreads. In this case, you probably want something that's going to replicate. But Taylor sins are, um, we think they're captured phage in some cases
Yeah.
where, or genetic mobile elements where the bacteria will produce these things that look very much like phage with deformed heads, and even sometimes they even package random bits of DNA into them, which is
Okay.
cool because again it shows you that
Yeah.
these were at some point phage that got captured and like neutered. They will then bind other bacteria, the same species as them typically, and kill them.
Hmm.
And so but they don't but they then don't replicate, so there's you lose that step where you when you're treating something like this you probably want it to replicate because it's everywhere. It's quite a big structure, so you want to get it going. So yeah, you'd have to find a way for phage to bind. Let's say that's trivial. Then you have to
Okay.
find a way.
Okay. Yeah.
Then you have to find a way for the once the phage is bound, that conformational change happens, so the phage has some way of inputting genetic information in. You then have to hope that the genetic information inside of this is there. Is actually programmable in some sense, because you could say that this wraith pathogen latches on, and then you, like as a virus does, it maybe it could use the host's own genomic information to replicate. It could cut
Yeah.
things up and say, well, we know humans have these genes, so stick them together. Um, this can show how nerdy I am about Stargate. The wraith are actually a chimeric species between humans and a bug, so they are part human, and it wouldn't be crazy to think that they'd understand the evolution that occurred, or the directed evolution in this case might be that they'd use their human parts of the genome and said, okay, in order to make this, we can just take these two genes, cut out the introns and ligase them together,
Yeah.
and now you have a new gene that will do this thing. But that the information in the pathogen doesn't have to include that genetic information. It can just include the parts that will just pick those things and do that. And then you could build a pathogen without having too much genetic information. So I don't know how a phage would get around and say, well, I have to use some DNA somewhere. I have to use some cellular process somewhere to make stuff. Where is that coming from if it isn't like a bacterial cell? The stuff in the show is, I think, human tissue effectively. It's
Yeah,
It's not she's not growing bacteria. She's growing
I have a lot of headcanon theories about this or hypotheses. We because we talked a lot on the when we talked about living ships, the problem of
yeah.
matter, of mass,
Yes.
how you're creating things like this. Sort of my pet theory for any problem of mass is simply that they are using pathways similar to plants, grabbing
Yeah.
out carbon and fixing carbon from the air.
Yeah.
That's why I think even just saying pathogen is maybe the more correct way than even bacteria. What we're
Yeah.
seeing, if we're using my head canon as sort of the basis, is part of this of a growing organism that isn't
Yeah.
bacterial in nature. But you're
It's
totally right. The wraith
basically a
are
tumor.
sorry, what?
It's basically a tumor. It's
Yeah. And the
yeah.
wraith, by the way, top tier genetic engineers. I wish they could give like the keynote talk at a conference.
Yeah.
Yeah, they're
So
fantastic.
and Target Advance has a lot because they have the other thing I got was confused with is they have another uh the vaccine or the treatment the gene therapy
The
for Wraith at the end that makes
yeah,
Michael like it's the idea is that they remove the non-human DNA from them and whatever the
yeah.
bug is called.
I would love the Aratus bug.
Aratus.
So I've already had a stream on this. Uh I have a so there's some like non-show like novelization
Yeah.
explanation for this, which I don't like the novelization. I just like
I've
the
never read the novels.
yeah. So in the show, they hypothesize it was bug up. I think
Yeah.
I like the idea of more that was human down.
Yeah.
If we're describing it, it makes a little bit more sense because then the transition form between a literal insect to a
Mhm.
wraith is a little less morphologically abhorrent if you're going from human to wraith.
Yeah.
Um, but anyways, yeah, no, and I actually I would kill to know the exact writer that came up with that, and I want to know what they
Yeah.
were thinking for gene therapy and what they were turning off, and because at that time CRISPR was unknown. There was no such thing as CRISPR. I think even TALENs and zinc finger nucleases were not a thing yet. I'm just wondering what they conceptually thought they were
Yeah.
doing. You know what I
Yeah.
mean? Because they had the right words, but they were like a decade a little too early for some of the
It was
difficult but still
yeah.
hard or
It was 2007 and 8, right?
for which
For Atlantis. So it would have been
oh
2000,
uh
2008 CRISPR was when I mean it's still after.
yeah, as a genetic technology, I think it was 2012,
That's
2013.
when yeah,
If they came
the
up with the idea like, oh, we can just have an inactive nuclease activity and target specific genes to turn them off. But yeah,
it's still yeah. Restriction enzymes are probably I
well
places
known. RNAi certainly was around.
RNAi, yeah, that was a big thing at that point, wasn't it? There was all the
Yeah.
stuff, yeah, yeah. It's funny, like you mentioned, it's like no, zinc fingers weren't that close. It's like yeah, they really were actually,
Yeah.
yeah.
So yeah,
Uh,
I'm very curious what conceptually they were thinking of, or if they just honestly, in thinking about it closely enough, they came up with the words that we would be using. Like, because certainly gene therapy, that idea was there, but
yeah.
I wonder what they thought that meant.
Again, somebody to ask David, like who was the right? Like, I'm sure
Yeah.
you might be able to figure out who it was, but it's a really good question. Like what again, this is where like scientists owe something to sci-fi because again, they clearly sat down and was like, what would be cool? How would
Yeah.
like we don't care how it actually works. It's just like what we term that. Something in the back of my head about there's been a case again where like iPads and stuff. It wasn't iPads, but where sci-fi has basically laid the ground. That we now call it that because of sci-fi. There's some good example of that.
Star Trek, the things they carry around are called pads.
Yeah,
They call, I mean, they're effectively iPads.
yeah. So David likes to say that when he was doing the SGA, they had these the handheld computers, which were the laptops. that had like touch screens on them. He's like, they gave him a stylus. And he's like, just basically threw it away. He's like, we're never going to be sat there with a stylus, dude.
Yeah.
It's going to be fingers. So like, you'll see him all the time. He's like tapping away at it. And again, that's
That was I hadn't realized
yeah,
that. It seems so natural.
before iPad. Yeah, yeah. But he's like, oh, you wouldn't be there because I think the computer actually had a stylus that worked. It was a proper computer that
Yeah.
had a stylus. He's like, this is terrible.
Wow, that's fascinating. I did not know that. Sorry,
It's a,
I'm
so
just catching up with the chat because I'm not ignoring you, chat. Um, uh, GNM doing stem cell culture in gene therapy lab vector studies too. You can do anything sci-fi. Oh yeah, you didn't like the second X-Files movie? I'll admit I never saw it. So
I've seen the first. I don't remember it.
yeah. Um, uh, you said something that was the perfect transition, and now I've forgotten it because I was really excited to have found this. So this is still Stargate, but moving
Yeah.
to Stargate SG
All
One,
right.
the Twilight years, as I would like to
Yeah.
call them, uh, when the priors sent off a virus. And I actually don't quite remember this episode nor what's actually going on. When I saw this, I recognized what it was immediately and looked away because
Yeah.
I didn't want to like spoil it for myself.
Right.
So
It's a plasmid.
exactly. So for anyone who does not know, what we're looking at is a report on Stargate prior to virus pandemic, an alien originating virus. And this right here on the left-hand side, I think you can see my mouse, is a plasmid map. So a map of DNA elements for what ostensibly is an alien virus. So I am now going to look at it because to see if I recognize anything.
So there's REI and RAT.
All right, well that immediately screams bacteria, right? Oh, and
Yeah.
TNPA. Oh yeah. And look up at the top, oh right. So it's TRBE, TRBF, TRBG. That's an operon. Like
Yep.
just, um this
Wonder
is
if they.
see this stuff I love. Also uh have you seen Neon Genesis Evangelion? It's an anime, very famous, but it's
No, I have. I know of it. I have never watched it.
every once in a while they flash on screen protein
Yeah.
sequences, and I
Okay.
just blasted them once. It was just random sequences they had clearly found and like thought were cool looking.
Yeah.
Okay, so I'm gonna look this up. I don't really know how it, I guess if you only know the gene names. Okay, because this is, how
I think
big
I
is
know get
this?
60,000 kb, 63K.
Yeah, so that's pretty big.
A big plasma, but it's about the same size as a phage.
Yeah. Oh, so maybe.
Yeah. The
Um,
thing I found interesting, they've got an origin and replication,
yeah.
but they don't start that at 0.
They just didn't start their plasmid map at the right
Yeah.
spot.
Yeah. It happens. It happens. All
Um,
the plasmids. Did you ever work with plasmids in minutes? Are you old enough to have worked with minutes plasmids?
no. What?
No, it like tail end. So the way we used to map plasmids was we do a conjugation experiment with like E. coli. We conjugate it across, have a stopwatch, and you'd say, okay, minute 1, stop the conjugation, break the conjugation apart, and figure out what gene had got into the
Ah,
bacteria. So and you'd
yes.
you'd
Yeah, yeah.
have this map wouldn't be in KB, it'd be in minutes. So you'd have like this gene is at this many minutes because it's
I
how long it took to get
I
across.
remember learning about this. You know what we do these days, at least in my lab? We spend $15 and get a complete full plasmid map of
Yeah,
anything less than, I think it's 75 kb using Oxford Nanopore. Plasmid Source, my new favorite company.
75 KB.
It's amazing. I
That's
think the upper limit is 75. Also, you don't need any primers. You literally just need a plasmid in a tube, send it to
Do you reckon
them.
they do a phage?
$15 later. Oh, I don't know. They do plasmids and linear amplicons of certain.
Oxford Nanopore?
Well, that's the technology that they use.
Yeah,
The company is called Plasmid Saurus.
Plasmid source. I like the name.
Oh, it's great.
Try to see if they want to take a phage because I got some phage which is like 60 to 75 KB. It's like,
Brian in my chat also apparently really likes Plasmid Saurus. Okay. TRBE. See, I just want to find out what this is. What random things did they Google to have found this?
Yeah, start
Oh
searching this.
wait.
It's
Oh
E. coli TBR gene. They've taken E. coli plasmid somewhere.
yeah.
Conjugate transfer protein.
I just found that same thing. Like PML123. I'm just wondering, is this just a plasmid or is it something?
It's quite big for a plasmid, but I mean, it's on
Or
like
like
the
a back
T.
mid or a back.
Yeah, the TRGB seems to be a plasmid transferring TRB system. Hold on, let's look.
There's
Uh,
also those.
is a TI plasmid system, so it
Yeah.
allows things to go across. So again,
And
the
there's some
CDELFGF conjugate transfer plasmid.
coloration, which makes me think, oh yeah, actually yeah, there's TRA, TRB, TNP. Those seem to be the major things. I just I'm looking for something unique and identifiable.
I
XF.
don't see any.
There's not a multiple cloning site, at least labeled.
Or yeah, no, yeah, or antibiotic genes are obvious. But
Yeah.
that might be a sulfur one there. I have to say it's a little short for a sulfur gene, but some big genes in this too. That's a long gene.
Uh yeah.
TRB is quite. It's just and TRAC.
So actually, there's a gene called XF2080, which I see there. It's right near, it's in the yellow portion by ORI5. That is a toxin.
Okay.
At least that's the name of a gene that
Yeah.
expresses a toxin.
No, I
Hmm.
didn't get the colors right. Like the yellows don't line up with the blues.
Oh yeah, the map is a. Oh yeah, I didn't actually even notice that.
At
Yeah.
least they got the Stargate SG command in the center.
Slapped
That's a
right
good
in the middle.
where you want that information.
Yeah. Um,
I was just reviewing a paper that has a plasma map. Let me just see what they put in the center. Because if they didn't put an SG logo in there, I think I might ding them for that. You know, they just put the name of the chromosome in there. Crazy.
let's
I
see.
can't. I unfortunately I can't show this, but I'm looking at a real version of this. One of these for a paper reviewing it. It's funny how close they are. I guess they must have just been like, let's find something. But I don't recognize
Oh,
this
for like
plasma.
a plasmid map?
Yeah.
You mean? Oh yeah, for anyone in chat, yeah, like this is. I mean, this is a little excessive. This is huge, but these are pretty common. That's why immediately when I saw this, I was like, I know what this. I know my brain knows what this is,
Yeah.
even if I don't know what specifically it is. Yeah. So I don't want us to have to necessarily sit here and figure this out, but I just thought this
Yeah.
was like hilarious. So this, oh, Pac M like. It seems like though this is definitely a bacterial
It
plasmid.
is bacterial genes, yeah.
It doesn't
They
look
are.
like it has anything to you, right? Anything
No,
phage.
this isn't phage. If it was phage, 90% of this would be hypothetical proteins.
Okay. Pac M like gave me Pac Man, even when I put the word biology next to it.
We also um phage typically, at least pseudomose phage, the what's the word, the synteny of the
Yeah,
genes in the
synteny, the order of the genes.
yeah, yeah, but they also they typically go in one direction. So whatever, like
Oh, they'll all like be
they'll
like
all go, on this strand.
yeah.
Um, that's it. It's far more common. So having genes that go back and forward, like on a reverse strand, isn't super common. So we probably wouldn't see it.
Oh yeah, I see. Yeah, you're right because these are flipping around all sorts of
Yeah,
different
I mean,
ways.
there are sections that obviously had some sort of inversion happen where they'll flip, but there's some thought that's kind of a recent occurrence when we see those, not it'll flip back at some point.
Yeah.
We're still not sure why, and the synteny and phages are really interesting. So for the viewers, the synteny is like gene A is next to gene B, next to gene C. We see that across different species where we see, okay, the gene that makes this kind of structure, we see it next to each other quite commonly.
Yeah. Hmm. I
Um,
see someone in the chat has some information. So doing some Googling on the side. So thank you, Watcher 652. You found it was on the Stargate Wiki. The document was in the episode The Four Horsemen. The JPEG that I pulled from was from eBay. So maybe it was something, it was an auction, an old auction of a Stargate prop. Just makes sense to me. I mean, I don't remember a close up on this from like any particular episode, so I'm not surprised.
It's
Also, but I
ionic.
do like this random text, by the way, just on the right hand side. Who knows what these things mean? At point of infection, one clone of the virulent alien infection,
Yeah, and I was trying to
VIAI,
look.
is already
Was it?
altered. Like someone put a little bit of work into
Yeah.
this.
I thought they were going to serve like some sort of homologous recombination of these because like why else have two chromosomes lined up like that?
Yeah, I'm looking in the random little bits of text. I'm
Yeah.
not seeing.
I also want to know what a cryonic forecast algorithm is. So is that actually a thing?
Also, that's not how you spell algorithm, is it?
It doesn't look like it.
I don't think algorithm has rhythm like rhythm.
Yeah.
Pyronic? I'm gonna look that up. That's forecast is also spelled incorrectly.
I got class. Yeah, I don't see it anywhere. It doesn't turn up. Time series forecasting models with Python. I meant Pyronic, not Python. Yeah, I don't think that's a thing.
Yeah, I got a pyruvate sensor.
Yeah.
Oh wait. Actually, pyronic is a gene, but in this paper from 2020, so I'm sure it's a fret sensor. I'm not finding any papers from back then, so yeah. Anyways, I'm sad that I've clearly like missed this prop by several years. As Watcher pointed out, it's too old. Probably like the eBay page doesn't exist anymore.
The
I think they do
dates.
those typos to see if anyone is paying attention. Well, it's a little
Maybe.
late, but we're paying attention. I mean, we're sitting here trying to figure out what these genes are doing.
They're implying that they're implying that there is some so on the chromosomal maps on the right, they're implying normal gene val, I assume.
That's
uh,
virulent alien
a
infection, VAI.
what's VAL then?
Where do you see an L? I think that's
On
just
the
an
left.
I.
No, so it goes from so there's normal VAL, a normal VAI gene,
Oh,
and then it goes down. It's like infected VIA.
I don't know
Yeah,
so those are clearly chromosomes. Wait.
yeah,
Oh, yeah. They're describing integration into.
yeah.
Wait, normal
Erratic random infection. Point of infection on one clone of virulent AL infection is already altered. Altered gene.
VAI gene. That I gotta look up human VAI. Just in case someone found a human gene.
Yeah. I do like their infection map as well. Like there's doesn't make a lot of sense in
Yeah.
terms of population density in places because like New York only has 632, Texas 128, 1,280, Oklahoma 519. So it's like, why
So
would Oklahoma have that many infections when New York doesn't? Has only about the same.
so hold on. Adenovirus has VAI genes that I think are inserted into human host DNA.
Yeah, I don't. I also, I, I think people worry. In case people worried, I don't like. I don't want to have a go at the prop people. I think that this is this is far more effort than they should have ever put in.
Absolutely no. I just like the writers. I applaud the fact that they're trying, and it doesn't matter that they're wrong to me, because like who cares if they're wrong? It's just fun.
Yeah.
But yeah, no, someone put some work into this. I wonder if VAI. It would just be so much fun if VAI they found that acronym from a real adenovirus, gene that gets inserted, and they came up with virulent alien infection.
Yeah.
I should search that. Alien infection. Just checking that out. Nope, that phrase does not show up. Okay, cool. Yeah, now I really want to. Yeah, sci-fi gal. Now I really want to rewatch this episode 2 in order to find this prop.
Wanted to watch Atlantis the other day. It's no longer on any of the multiple streaming services I have, so I was all like, put out.
Is it not on Hulu anymore? I was.
Is it also? Oh, I didn't check Hulu.
Oh,
Yeah,
I was gonna say
already.
I was about, I want to say, halfway through one of my many rewatches, and I just kind
Yeah.
of put it down in favor of other things. But I really wanted to finish it. SG1 Netflix, SGA Hulu. Thank you.
Okay.
Good
I
information.
thought I was looking on Amazon, Netflix, and Prime. Sorry, and HBO. Oh, I've got Hulu,
Yeah.
so I'll have to watch some stuff later.
Oh, and Pluto TV.
Pluto TV.
Yeah.
Thank you, chat.
And if this episode has commentary, someone might say something. I'm such a bad fan. I never try finding commentary for things. I really should, because you're right. There could be
That's like
info there.
the Rick and Morty commentary. I
Yeah.
can remember I was watching that, but they were asking questions about the human body and choices that were made.
Exactly. So I also wanted to mention, just because it's 10. This 10 is usually when I'm like, okay, when's it time to finish for the night? I'm happy to keep on talking. This is like very exciting, but also I know you were streaming before. Long day. If you're done, we can be done.
Well, we can do one more topic. I'm happy
Okay.
to do one more.
Okay, great.
Definitely. Yeah.
Also, Derek, VA or VAI is just denoting viral associated RNA. So that makes me think that where's the I come from though? This person would know if it's something medical. So tell me where the I comes from, and then I will believe you. Interference. Okay. Interesting. I'm more willing to believe that they found that acronym and then pointlessly, because who would find this except for us now, change it to virulent alien infection, which is wonderful. All right, so one more topic. All right, well, we can talk about something that's not on here, but the only 3 slides I have left are the macrovirus from Star Trek Voyager, which itself just has some interesting ideas on a different type of biology, I guess. There's the X-Files Black Oil, which is a little bit more mysterious, and maybe because of that has a little bit less to sort of think about. And last is just the general concept of a zombie virus. So, do you have any opinions or thoughts or anything like that?
I remember watching the Voyager episode about the macrovirus. That's a cool one. The other and the zombie stuff, of course, the classic one is rabies, right? Which I know a fair bit about. But the sci-fi here. This is one of the worst parts of sci-fi when they lie by mistake, where they say something that doesn't make sense.
Sure.
They say something along the lines of the DNA has been incorporated into its amino acids. I think that's one of the lines.
Yeah,
in the show.
yeah.
And it
hydrate.
It doesn't. You don't need to say that. Like, you just don't say those words. There's a classic. I think it was Prometheus, the movie where they were like, "Oh, with carbon dating the planet, it's this many years old." It's like, that makes no sense. You have no idea what carbon 40. Like, you don't know those things. Useless. Just take a machine out and go, beep boop. My science machine says it's this many years old.
When they,
Don't what
yeah.
like they stretch into words that actually have meaning, then this doesn't work. It's like, wow, you
Um,
could have asked a scientist 5 seconds.
so
Does this make sense?
my mod Brian just found the quote: "The organism had adapted to incorporate the growth hormones of its host into its own amino acid structure."
Yes. Yeah.
Sort of like trying to
But
link to that's what we're looking at right now. So if anyone hasn't seen this episode, they get the crew get infected with a virus, which I think is already quite macro because I think they can hear it buzzing like a small
yeah.
insect, but they can't see it. And eventually, it grows to this size where this is a human right there, and it's still trying to infect people, but also people don't die. Like they don't do the like David Cronenberg version of this episode where people burst open after being filled with these giant viruses. They just kind of grow outside of the cell or of the body, not of the cell, of the body.
I do like the sort of geometric shapes the
Yeah.
virus has here. It looks viral. It looks like if I had to make a macro virus, it might look like something like this. It's a case of like, well, how would you, if you just have proteins, how do you build it to make it strong? And there's corrugated cardboard, basically that probably is. Let's be honest, it probably is corrugated cardboard they just made on the prop there.
Well, I think this was all
Super strong.
CG actually.
Oh, was it? Wow. When was this? When was that out?
Voyager
When
was
this?
pre.
Yeah,
I
as a
think
kid.
like someone's gonna look this up. Pre
Yeah.
Enterprise, so pre early 2000s, but
Yes.
mid and post DS9. So I swear it was like
Yeah.
97, 98, 99.
It was DS9 kind of had its end as this was starting, right?
Yeah, and the thing with Voyager is that it's oh wait, hold on. Sci-Fi Gal has to leave. Thank you for coming. Yeah, so Voyager is the first Star Trek series that used like I forget what the like metric is, a lot of CGI. So they didn't have they don't have a model of the ship or any ships,
Okay.
and they were actually relying on CGI. They also like were the first to not the first. I think they mistakenly like it wasn't good in retrospect, but I'm pretty sure that they filmed it all on like tape.
Digital.
No, not
Yeah.
tape. Or
so tape or film. So if they filmed it on tape, it would look crap nowadays. If it's filmed on film, you can blow it up now.
So I think it was on tape because all the others have been remastered because they were all on film, and I think Voyager got the short end of the stick and they put it on tape.
Yeah.
So we've got multiple answers. December 11th, 1996. All right, so way earlier than I thought.
Yeah. Well, it's like Star Trek Next Generation. I think it's only a few years older, a few years younger than me. It's like, oh shit. Like it started in the 80s. I was like,
Yeah, no, they all happened like in overlapping times, I think.
Yeah. But so I was saying, like, if I was to design a macro virus, it's like simple structures where it's like, okay, well, you want something strong. These geometric shapes of like corrugated cardboard would make sense. The problem becomes energy again. Like, how is this? If this is a virus, how is it metabolizing?
Yeah.
At what point does a virus just become a predator that if it's just eating you and are those internal parasites, are they viruses just because yes, they have to live inside you to replicate and grow, but there are obligate pathogens that are like macroscopic.
Yeah, they kind of missed the boat on sort of like they were trying to make it sci-fi and they were trying to connect the ideas of growth hormones. I think that's where they latched onto was like if they absorb the growth hormones while they're in you, then they will grow when they're outside of you. But the problem of the mass, that's a problem a lot in sci-fi is like, where's this coming from?
What's like the xenomorph in Alien? Like, how does it go from yay big to 8 foot long in a short time window.
Oh boy.
Yeah.
Did you see this quote that Brian put in about how they so Janeway contained it in one area and threw an antigen bomb at it.
I mean, that would work. An antigen bomb would
I mean, it would work. Yeah.
just got us there. Um, what is an antigen bomb?
It's
Like,
not,
is it just shrapnel of antigens? Like, it's
uh,
just a grenade at that point,
yeah,
right?
that's what it sounds like. I mean, I would oh yeah, and they confuse the virus with VR. Although the holodeck is special and can simulate like sense and sort of things
Yeah.
like that. So if it's following a molecular trail to its hosts, I could see that sort of being able to like utilize the holodeck for that. But yeah, no, you're right. An antigen bomb. Also, that seems like trying to kill Godzilla with arsenic. Like the problem is not necessarily like killing it dead. It's also or like stopping it. It's like the mass
Yeah.
behind it. Like okay, so it can't infect you, but it's still the size of like a basketball. Like it would still probably do some damage if it just
Look
ran
at
into you.
you. It's
I mean, they got the. Wouldn't you say that they got the words right? Like it's kind of like you were just, or maybe not the words right. What I'm assuming that they mean is that they are blocking its ability to infect a host.
oh, so like they opsonized it in some way with
Sure, yeah.
antigens that then it can't uh, find a new attachment.
Now, if that worked on the microscopic scale, sure, but again, I mean, why would that would work on something so huge and make render it ineffective?
It yeah the that only works if it has to be internal. If it's mechanically entering your body,
Which
like
at this size, it kind of seems
yeah,
like it would
it
be.
has to. Yeah, it's not an it's not a parasite like an intercellular parasite at that point.
So we got the link to Memory Alpha, which is simultaneously the most useful and least useful wiki out there. So it is an antigen bomb is you constructed by attaching a canister of the macrovirus antigen to a grenade. So it's yeah, what it is right on the label.
It's like you could have any kind of
Exactly. She could have just probably
grenade.
thrown the grenade and been okay.
Yeah. I don't. How many other grenades are there in Star Trek? Like, when else do they use grenades?
Uh, so they use like stunning grenades every once in a while that I've seen, uh, but not often. And sometimes they do like sort of hand wavy like particle grenades. It's very loose definition.
Yeah.
The only time they ever use explosives often is when they're using like matter antimatter collisions, which
Yeah.
are explosions on an order of magnitude so far and above what you would need to incapacitate an enemy that I don't think it comes up that much.
Yeah. It's just one of those things. Like, there's times when a grenade definitely would have been useful in Star Trek, and they just don't use it. And yeah. Inconsistent, but it's fine. It's fine.
Yeah,
It's
yeah,
fine.
no. And
Yeah.
Watcher is pointing out that this thing must be really weird since the transporter filters let it through when the doctor, a holographic being, beamed down to an infected planet and brought it back with him when he beamed back up. So they beamed up just his holographic generator and virus somehow. Yeah, that's
Well, the um, transporters basically are. If they work as they work on the show, the Deus Max machines, like they do whatever you want because they can reassemble matter. Like,
more or less, yeah.
if someone's dead, you could put them in there, scan them, and then tell the machine, well, actually, like you know what a living machine, you know what a living human looks like. Just put them back like you did last time.
Yeah,
And,
there's yeah, exactly. There's some philosophical problems with
yes.
that.
I think
They do get. I don't know if you have seen or own or read, but they have the Star Trek: The Next Generation technical manual is a
I've seen
wonderful
it.
show used document um, where they outline all of the sci-fi technobabble.
Yeah.
They get around it by saying that the transporters, uh, they scan and recapitulate quantum state. I think what they're trying, because the what they're trying to do is not kill you every time they put you in
Yeah.
a transporter. And I think they're basically saying that the soul and life exist at the quantum level, or at least what we care about it exists at that level. And if
Yeah.
you can copy that perfectly, well, then you're fine. Um, yeah, no, the teleport is its own thing.
If they get a physicist for that one.
Yeah, no,
This is-
well, or a philosopher. See, that's the thing.
Yeah. There's
Um,
I think it's CGP Grey YouTube videos he did about how teleporters are just suicide machines.
yeah, I think I remember that. I mean, I have my own thoughts on that, which is I almost agree with the Star Trek technical manual, only in that, like, when I think about it, and obviously it's not the same. Obviously, it's like a measurable phenomenon, but from the perspective of the person who's experiencing it, what's the difference between transportation and going to sleep for 8 hours? Either
Yeah.
way, you let's set up an experiment where I am transported by an alien civilization from my bed to my bed while I'm asleep. Did I die? Does it matter that I died? Like, you know what I mean? It's yeah.
When
So
my Doctor of Philosophy failed. Like, not that kind of philosophy.
yeah, yeah, exactly. The P in our PhDs
Yeah.
or PHs and PhDs aren't
Yeah.
doing much for that conversation.
So it's I'd want to say like, oh, it's something we never have to worry about, and it's always like, well, this sort of quantum questions, our understanding of quantum dynamics has a point where we can entangle things across distance. Like, okay, well, the reality we exist in is like a thin smear of what reality actually is. Our
Yeah.
ability to perceive reality, I think, is skin thin compared to what the universe is actually doing.
Oh, absolutely.
And it's like, well, maybe we shouldn't think about that. Maybe we should just be happy in the sunshine,
And
relaxing, and then we don't have to worry about the void or yeah.
exactly. And perhaps you're aware of or even read the paper where a group quantumly entangled some tardigrades.
I've heard of it. I haven't looked it up. I recall it being someone saying it was less impressive than I thought it was going to be.
It's kind of stupid. It's basically
Yeah.
that we can quantumly entangle matter. We
Yes.
quantumly entangle matter by making it cold.
Mhm.
Tardigrades can survive the cold.
Yeah.
Therefore, we've quantumly entangled tardigrades. I mean, they did it, and it's cool. And the tardigrades survived, but they more or less just survived being super
Very
cold.
cold. Yeah.
Um,
That's
yeah. So, and then now people are bringing up more, more of the soul sucking reality of what
yeah.
teleporters could be.
Scotty was in the replica. You know, they explained how that's why he got old because the pattern degraded slightly. But that was the Dyson Sphere episode.
Yeah.
Um, yeah.
Great episode that skirted around a lot of cool stuff and then never was revisited.
Never once go like. Remember that Dyson Sphere we found? That's clearly like a class 4 galactic civilization exists.
Exactly.
Like,
Let's not investigate it.
let's just leave that
Let's
as
spend money on a perfect reproduction of the original TOS set, bridge set. Yeah, no, that's I mean, I don't know if you're watching any of the new Star Trek.
I haven't yet. I keep meaning to like because it's on PBS or something. It's on some streaming service I don't pay for. So
Actually,
like,
which one do you think I'm talking about?
there's the current brand new one with Captain Pike, which I've heard good things about. There's Discovery, which I haven't watched, and I've watched a couple episodes of the cartoon Lower Decks.
Lower Decks
Lower Decks is
I think is
hilarious.
hilarious. Strange New Worlds is the new Pike one. It's really worth watching. It's
I heard it returns to that sort of like philosophy of Star Trek where it's like, well, maybe we should have a discussion about this. We're not just pew pew Abrams style machine gun wielding super soldiers,
Exactly.
where actually it's
There's a
yeah.
character in there who is repeatedly stored for medical reasons in a transporter
Okay.
buffer, which is interesting and also quite horrifying. Very
Yeah.
horrifying. But yeah, my I play a Star Trek based RPG with
Yeah.
some friends. We spend more time in self organized committees than actually moving the plot forward. We have so
This
many
is what the Federation is. That's what
exactly.
it's,
Our DM, who may or may not still be listening, actually right now.
yeah.
Recently, we were uh, meeting a new race. We were helping some diplomatic things. He made an off comment, sort of description of how they greet each other by holding each other's like foreheads and giving their heads a little squeeze.
Yeah.
And we talked about that head squeeze and the subtleties of it for like 15 minutes, just to make sure our fake characters did it
Yeah.
right.
Did it correctly. Yeah.
Yeah,
That sounds like a true Star Trek experience. I was gonna say I enjoyed the Abrams films
yeah,
because they were actually fun films, but yeah, it's uh, giving him more science fiction properties is not a good idea. Uh,
yeah.
Again, saying that I love Fringe. He was involved in Fringe. He gave a TED talk
Hmm.
decade ago now, I guess, and he kind of outlined his philosophy for making shows, and it's like you dick. like, like
Oh wow.
his entire point was never give them an answer. Never tell the audience anything
Oh, I've seen
useful.
this. Yeah, sorry.
The box.
You
There's a secret in the box. You just keep giving them smaller and smaller boxes.
You know, you said J.J. Abrams. I heard Simon Pegg, who I know like was
Oh
involved
no.
or helped write, and I was like, oh
Yeah.
wow, I didn't. No, JD. I remember seeing this. Yes, you're right. That's the worst way to write. I mean, stories, but also sci-fi. I mean, the fun
Yeah.
part about the sci-fi is you can open that box and be
Yes.
like, here's a basketball-sized virus. Who cares? Like,
Yeah.
you know what I
Yeah. Yeah.
mean?
Well, because he did it with. I think it was built on predicated on Lost, and I watched the first season. Lost was like, oh, that's purgatory, and then never watched it again. And then people were like, no, 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 they've told us it's not purgatory multiple times. The showrunners have confirmed it's not purgatory. It's like, oh, it turns out they were all dead. It's like, okay, they built up a show to such a high level. It's like if you just take a step back and say the audience is allowed to know critical information, that's
Yeah.
fine, as long as the characters have different expectations of different modes of understanding. Star Trek did this great, right? Again, like go back to Star Trek, where the Federation is more or less uniform in how they believe diplomacy, science, warfare, all these things should be handled, while the Ferengi are like, how will we make money on doing this? How is this profitable? How do we how can we make war make money? How can we make slavery? How can we do that? And it's like when they butt up and say, okay, well, this is they have the same information. They're not, no one in that universe doesn't understand that an economy exists, but they have different modalities to get to that point.
Yeah.
You can do shows where people have that information. I think J.J. Abrams is very much like, again with the Star Wars, it's like, oh god, like his movies are okay, like they're well made, they're interesting, and it's like, why would you give him a trilogy when he's famously failed to deliver on any
Yeah.
story more than one episode long?
And they did it in the worst way. I do feel bad
Yeah.
for Ryan Johnson, who I admit
Yeah.
I did not like his movie
Yeah.
until I saw it in retrospect.
Yes.
Until J.D. Abrams, as you said, was given the
Finished
reins
it.
of the last one,
Yeah.
and he did try to answer things, but he also he yeah, I think it was a good setup, but then they like did the mistake. They had J.J. Abrams, who's good at setting up questions, not answers. Ryan
Yeah.
Johnson comes in, subverted our expectations. I don't know who should have had that last one to hit it out of the park, to like put it all in perspective, but it wasn't J.J. Abrams.
Yeah. No, it wasn't. He didn't want to be. I'm talking for him now. Like, why not? He didn't want to be pinned in by the narrative that had been built by Johnson. Like, but then like so much stuff in that third movie was like, what? why? Like, I do not like the prequels, but even now I'm like, they might not have been as bad as those. Like, I grew up. Star Wars was, you know, A New Hope, Strikes Back, and Return of the Jedi. And then I saw the prequels when I was, teenage years. It's like, they're okay. Jedi's are cool, I guess. You know, they've turned from like mystics into, I guess, yeah, Jedi Knights now, which are kind of influenced by the games. Like, there were a lot of the games coming out, like the Carl, is it Carl Catan? Is that the guy's name in the games? Dark Forces and stuff like that.
Yeah.
Jedi Knight games. But the Abram, like the reveal of like basically an entire new order of battle cruiser ships, like what the hell? Like, who built them? Like the crazy
Force?
thing is also that yeah, the Force made them, I guess.
Yeah.
Yeah. The expanded universe, the galaxies as it used to be called, like, but they had all, Outer Rim stuff. There's a lot of cool lore there and stuff. A lot of fun. Like, fans absolutely love it. Again, I haven't really gone into that deep, but it's like there are stories out there which they could have just said, yeah, they're part of the Disney. Disney owns these now. Let's work with those instead of end up with a really bizarre mishmash of ideas and not really explain to us, well, how did the Emperor, like, what? How did he fell down a chute? Like, is that the same guy who fell down, or is it a clone of himself? If it's a clone, why is he so fucked up? Like, so many questions.
Yeah, no, I mean, the problem is, like, I think it's, how do I say this? It's okay. It's okay. No, oh, no. It's okay to, like, have open-ended questions like that don't need to be answered. The problem is that they were all presented in a way that kind of makes us want to know why. And that's the problem. There's like a certain set of things in sci-fi where it's like we should just be able to say this thing and you accept it for what that face value. But the way that the newest movies were presented is like there's so many things being answered unnecessarily. How Han Solo got called Han Solo. Like, why does that matter? Who cares? That wasn't even like,
It's just his name. It's a cool name.
Yeah, it's unnecessary, and it causes audiences to want to know more when really an audience probably shouldn't need to know more because we can't know everything.
Yeah. Well,
I mean, that's the downfall of the Dune series is Brian Herbert's son trying to go back and like explain every little bit from the glossary at the end of the original Dune book. But
yeah, it's just a good story about a guy finding Muad'Dib and what do the Jesuits call him?
The path between something.
Oh yeah, he has Kwisatz.
Oh, the Kwisatz Haderach.
Kwisatz Haderach, yeah. I'm listening to the audiobook because I read Dune. I tried to read Dune fully and gave up, and I was like, I'll listen to the audiobook.
I know people say it's sometimes boring, but I find it extremely engaging. I love Dune. It's the only book I've read like many multiples of times.
I didn't find it boring. I felt like I was like, I want to consume this and sit down. And then I was like, I got an Audible subscription. I was like, oh, I could just listen to it. Same with what was it, Ray Bradbury, Fahrenheit 451. 451. Like I say, it's something I should listen to. I should read. I should comprehend it. I have never done it. I was like, oh, I get an audiobook and now I can do it again.
Yeah.
But I remember trying to read Dune when I was younger, being like, I want to do this, but I don't have the time because it's Children of Dune and everything else. Someone was telling me about the is it Paul's grandson who turns into a giant worm, or is that his son?
That is his son, Duke Leto the second.
Becomes the emperor for like 4,000 years. I was like, that sounds like a really cool story that I want to get involved with.
And the reasons why he did it are also fascinating. I love such a weird thing. I love the end of that book.
I I enjoyed it. Really enjoying the audio version of it. So it's
I
it's done really well.
it's a sort of precipitous drop, I would say.
Yeah.
You'll find out if you decide to listen to or read to the sequels, you'll
Yeah.
figure out when it's time to stop
It's good
naturally.
to like how much of a more history of Dune do you need? Um,
Yeah.
the what was it? There was no the thought is gone. There was something in the back of my head. I was like important about. Oh, of course the 80s version. I watched that when I was a kid as well, so I think that also put me off investing in Dune. The Sting
Oh,
one
yeah, yeah.
made by what's the guy's name? Made Twin
David
Peaks.
Fincher?
Lynch.
David Lynch.
David Lynch. Just an amazing film, like terrible, like
So that
just done absolutely the worst way, and not really his fault either. I think there was like a decision for him to cut stuff and what to keep in and what go. But then again, reading the book, I was like, oh, actually, the stuff in here that he lifted directly from the book, like the narration is just like, okay, well, how do we tell the audience, the viewer, that Paul's going to go to Arrakis, or have them
yeah.
watch like an informational? It's like, oh, that's in the book. It's like, oh, I didn't realize that was actually in there. So it's a very
Yeah,
clumsy,
like the princess
visual way to tell people.
the Princess Aurelian quotes
Is-
and then the
Yeah,
glossary at the
her poem
end.
book and stuff. Yeah,
Yeah,
yeah.
it's and how you I don't know if you've seen the newest movie. The newest,
Yes, I have. Yeah.
absolutely love it. Like I
It yeah,
loved it
that was what
so
got me to go
much.
back and read it or listen to it again. It's like, it's a good story,
Yeah.
and
Uh,
I've got this audiobook thing. I might as well try to use it.
It's I mean, my Dune book is literally falling apart. I've flipped through it so
Yeah.
many times. It's such a good series. It's on my growing I have a list of topics, sci-fi things, sci-fi properties that I want to get to. It's a growing list. It grows rather than shrinks. Dune is up there. I just don't know what specifically to talk about. The biology of the sandworms,
Yeah.
Shai Hulud, or just the fact that half of the book is about ecology and
Mm-hmm.
terraforming. That's
Yeah.
amazing.
There's an imperial ecologist.
Yeah, that's a main character.
Yeah.
Imperial
Uh,
ecologist. That's a great job title.
it's a good title, isn't it? The I you got to talk about the worms.
Yeah, the
But
fact.
also
Yeah.
the
Oh,
lack of aliens and yeah, the navigators. I think a lot of people think of that they're aliens and stuff. It's like, no, they're just meta humans.
yeah. It's yeah. They definitely. It's a interesting universe because it's an empty universe.
Yeah.
But also technically, original Dune doesn't quite. They don't know of their history that much. It totally. I don't know the expanded universe past the first couple books. Basically, what his son wrote. I don't know what he wrote, but there's the potential, especially the way that humanity is sort of laid out as intrinsically imperialistic, not just in the structure of their government, but in the structure in how people conduct themselves as just it's actually a really sad portrayal
Yeah.
of humanity, but like intrinsically violent and genocidal. It would not surprise me if in sort of like the headcanon history of the universe, humanity spread like a plague and killed
Yeah.
off like any life forms that they came across that
Well,
were useful
there
for
was
them.
the thinking machines were all
The,
the part of it when they
yeah, from
destroyed all.
the Butlerian Jihad, the
Yeah.
when the thinking machines attacked, rebelled, were beat down by
Yeah.
the humans, and forever eliminated from human culture.
Yeah.
That's extreme. Oh, someone changed my lights. They're very bright. I'm going to turn them down.
It's a risky game to play, having people control lights.
Well,
I said I was going to mention this earlier. I have like Philips Hue lights and the Amazon control lady, and my computer obviously also has RGB and stuff. So I was playing around trying to make sure the RGB was going to sync with the temperature stuff. I saw a thing like, oh, you can link up your Hue lights. I was like, that could be kind of fun. Let's see how that works. So I linked them up, and every single 90% of the lights in my apartment are smart light bulbs. every single one of them started strobing, and like sort of 2 hertz sort of strobe on and off. And it turns out either like I tried to stop the thing, turned off the program, didn't stop the lights, turned off my computer, didn't turn the lights off, turned back the computer back on, everything's still strobing at this point. Find like Googling, like the only way you can unentangle the system is to go onto your like Hughes bridge, like directly SSH into it effectively, and disallow the RGB controller on your computer access to it. So I was like, for 10 to 15 minutes, I'm like, oh
Oh
god.
my gosh.
Like, I didn't have to like nightmare trying to turn off these lights. And you look up like online, like Q&As of people like, why is this a thing? Why can you not just say turn it off? No. As soon as you connect it, it's like it takes over every light and just does whatever pattern the computer was doing. Obviously my computer at that point was like doing a breathing RGB thing, so it was up and down, up and down. So every light did it no matter what happened. And like, I think you're brave to let people control lights.
Well, so my viewership is low enough that has not been a problem, but yeah, it's completely controllable via my chat. I love it. I love when my lights change. I don't know how to describe it other than like, yeah, I have my personal controls right here.
Yeah.
I can change them. There's shortcuts that I've programmed in as chat functions, but also you can just change it to any 2 RGB values that you want. I say when my average viewers start getting, it was already at that color, Twitch guy too. It just got brighter for some reason. When my viewer count ekes a little bit higher, like more stably near like 15, I will probably have a master off switch if it ever gets to be a problem.
Just starts. You can use there's channel points as well, stuff you can use it too.
Yeah, so I currently have a channel point thing where if you want a custom color,
custom light.
Yeah. So my mod Brian, but Icefire has a wonderful like set of lights. I really like this one. It's, I love talking about stuff, but my side favorite thing about streaming is messing with my light controls.
Hydrate.
Oh. Open my 3rd can of water. All right. Well, I feel like we're coming to a natural close. If it feels like that to you.
Well, my voice is starting to go. So.
You what?
My voice has started to go. So
yeah. Because how long were you were streaming for like an hour before you joined? Maybe an hour and a half?
I was doing some loud talking in the lab before that as well.
Yeah. Yep. Just talking at lab, at work, that also takes you out of it.
It's just so noisy in the moment. People renovating our buildings, so there's lots and lots of noise.
Yeah. Thank you, Brian, for reminding everybody to go check out James's channel. I don't know how to describe exactly. You've been playing a lot of, when I've been catching your streams, you've been playing video games. I don't know if you have like a current, like a certain thing that you go for.
Not really. I just I occasionally take the camera into the lab and do like I've done like some qPCRs and camera and stuff like that, and showing people what I'm doing in the lab. I want to do more of that. It's just been very noisy at work lately with all the renovation, so it's been a little difficult. I plan on doing that more with my new lab. I'm actually looking at different mics I can use that would cancel out some of the hiss that typically occurs in labs. But I also play D&D with some folks.
Right.
We're maybe playing on Sunday. We'll see. And yeah, if I feel like playing a game, I'll probably stream it. Or I think I'm playing Planet Crafter. Is the name of
yeah, Planet something like that. Yeah.
Yeah, which is a fun little game. So a whole bunch of stuff. But...
Cool. Awesome. Yeah. So go check James out. Otherwise, thank you for all the new followers, new viewers. I've seen quite a few new names. I'm sure a good portion of you followed James from his stream, but also welcome to anyone who just happened to wander in. So I guess quick I don't know, announcements. Sunday, I'm just doing my normal office hours. Not sure what. I might take a break from the microscope until I get something more interesting to look at. I've been playing a lot of Jurassic Park, so maybe we'll play some Jurassic Park. Otherwise, yeah. I don't know what to say other than thank you for coming. This was awesome. Maybe if there's another topic that I can think of or you're interested in talking about, we can have you on again sometime in the future.
Pleasure.
But this was amazing.
I want to bring David in just because I know how much he enjoys when two scientists sit and chat around him. Like as weird as that sounds, like a couple of times that we've had chats with a mutual friend of ours who does like high performance computing, and he was just on cloud nine, like sitting on this fly on the wall of this chat. So I'm sure he'd like to join at some point. I'll bug him into that.
Yeah, no. Yeah, that'd be great. I unfortunately he streams every time while I'm like in the part of the day where I'm being productive at lab. So I'm like surreptitiously like opening it up and putting it down so I can kind of listen for a little while. But yeah, people are saying thank you for coming.
My pleasure. It was great. It was real fun.
Great for having you.
Yeah. I'm glad I can make it.
Yeah, definitely. So I am going to say goodbye and then switch to my sort of finishing screen. Wow, can't talk. So bye everybody.
There you go.